We have got a massive, just a staggering stack of research on the table today.
Oh, yeah, it's dense.
Very dense.
Yeah.
And it represents this profound paradigm shift in how we understand our own biology. We're pulling from this vast landscape of literature today, everything from the latest updates out of the NIH Brain Initiative to these highly specialized molecular biology papers to clinical psychology reviews.
Reviews that fundamentally redefine mental illness, actually.
Right. And the singular focus of all this disparate data, the reason we are doing this deep dive today, is you, the listener.
Specifically, those 3 pounds of highly metabolic tissue sitting inside your skull.
Exactly. Because for decades, the dominant metaphor in pop science has been to treat the brain like a computer. Like it's this static piece of hardware,
Yeah.
a silicon chip where we just upload software in the form of memories or skills or daily habits.
Which is a terrible metaphor, by the way.
It really is.
Yeah.
Looking at the sheer volume of structural imaging and molecular data we have right here, that metaphor isn't just slightly inaccurate, it's completely inverted. The brain is not a static machine at all. It is a living, continuously shifting physical landscape. So the core question we are unpacking today is how our daily experiences—
Mundane stuff, really.
Yeah, the chronic hum of modern stress, the exact macronutrients we eat, the physical exertion we endure, the chemicals we ingest. How do these inputs not just change our mood, but actually physically rewire the microscopic architecture of our minds?
It's a complete redefinition of human biology. We are diving into the universe of neuroplasticity today, but we are moving way beyond the usual pop science buzzwords.
Heavily.
The literature we're synthesizing frames the brain as an organ entirely built around ruthless adaptation. So our mission today is to explore the granular mechanisms of how the brain alters its physical structure to survive its environment.
And we're going to look at the dark side of that, too.
We have to. We'll look at how the brain's attempt to protect itself can lead to severe structural degradation, things that manifest as deep depression and intractable addiction.
Especially regarding dopamine, which we're going to get heavily into.
Oh, the dopamine research is wild. And critically, we'll unpack the emerging science of how you can actively intervene in this process, how you can leverage pharmacology, neurostimulation, and specific daily protocols to hack your own neuroplasticity and build a more resilient mind.
Okay, let's unpack this, because the idea that learning a new skill actually changes the physical weight and shape of the brain is just mind-blowing to me. How does this actually happen at the micro level?
Well, the literature frames it around 2 main forces: long-term potentiation and long-term depression, LTP and LTD.
And these aren't just simple on and off switches for memory, right?
Not at all. They are incredibly dynamic, competitive forces constantly sculpting our neural networks. If we look at the synaptic level—
Microscopic gaps between the neurons.
Right, those junctions. LTP is the mechanism of encoding. When a presynaptic neuron repeatedly and effectively fires and stimulates a postsynaptic neuron, that connection physically strengthens.
The receiving neuron basically bulks up.
Exactly. It increases its receptor density and becomes physically more sensitive to future signals. This is the absolute cellular foundation of learning.
But the research really highlights LTD, long-term depression. And I was struck by this because it's usually casually dismissed as just, the brain forgetting things.
Yeah, which is a massive oversimplification.
Right. The molecular data shows it's actually this aggressive, vital survival mechanism. It prevents the network from just getting totally saturated.
That is a crucial distinction, because if a neural network only had LTP, if it only strengthened, the system would face catastrophic failure.
It would just overload.
Runaway excitation, metabolic exhaustion, and eventually excitotoxicity.
Excitotoxicity.
Yeah, where the neurons literally fire themselves to death. So long-term depression is this activity-dependent reduction in synaptic efficacy. It actively prunes irrelevant connections to maintain synaptic homeostasis.
It's like a city grid. Traffic, or neural activity, dictates which roads get widened into massive multi-lane highways.
That's the LTP.
Yeah. And the neglected dirt roads just get abandoned and paved over. That's the LTD.
Yeah.
It ensures the network stays efficient.
Exactly. It prioritizes the pathways that matter for survival and degrades the ones that are merely noise.
I found the mechanics of how the cell actually initiates this process fascinating. Specifically regarding the NMDA receptor. The papers describe it essentially as a molecular AND gate, a coincidence detector.
It's a marvel of evolutionary engineering, honestly. It's a glutamate receptor sitting on the postsynaptic membrane. But unlike standard receptors that just open up the second their neurotransmitter binds to them, the NMDA receptor is gated by a voltage-dependent physical block.
A physical plug.
Yes. At the neuron's resting membrane potential, the pore of the NMDA receptor is plugged incredibly tightly by a single magnesium ion.
So even if glutamate, which is the chemical key, binds to it perfectly,
The channel remains completely blocked. It requires the postsynaptic membrane to undergo a significant electrical depolarization,
and where does that electrical charge come from?
usually from neighboring AMPA receptors getting activated at the exact same time. Once the internal voltage of the cell rises enough, the positive charge forcefully repels that positively charged magnesium ion. It literally ejects it from the pore.
So it requires both the chemical key of glutamate A and D, a strong electrical surge to blow the magnesium plug out.
That's the coincidence it's detecting. And once that happens, the data shows a massive influx of calcium ions flooding into the cell.
Which acts as the ultimate biological trigger for construction, right?
The pivotal moment, yeah. Calcium is a powerful secondary messenger. It binds to something called calmodulin, which then activates an enzyme called CAMKII.
The molecular construction foreman.
Perfect way to describe it. It triggers this massive intracellular cascade that leads to the rapid insertion of more AMPA receptors into the membrane, and even changes gene expression to physically enlarge the dendritic spine.
That single calcium flood is the brain's physical way of deciding that a specific connection is vital and have to be reinforced.
Exactly, and that structural reinforcement maps perfectly onto Hebbian plasticity.
The old axiom, neurons that fire together, wired together.
Right. But the literature highlights a massive vulnerability in Hebbian theory. If you rely solely on firing and wiring, you create an endless positive feedback loop.
Because the connection gets stronger, which makes it easier to fire,
Makes it even stronger.
leading to that runaway excitation you mentioned, the excitotoxicity.
Right. And this is where the concept of metaplasticity completely changes the picture.
Metaplasticity, the plasticity of plasticity.
It's one of the most elegant regulatory systems in our biology. The brain has these built-in homeostatic mechanisms that continuously adjust the threshold required to induce either LTP or LTD,
Based on what?
based on the prior history of that specific neuron's activity. If a neuron has been highly active, metaplasticity raises the threshold for future LTP. It makes it harder to strengthen connections further, while lowering the threshold for LTD, making pruning easier.
Wow. And if it's been quiet?
The system makes LTP easier to achieve. It's a sliding scale. It ensures the neural network always operates within a safe, functional, dynamic range to prevent structural instability.
Now, the raw material driving all this physical growth, the actual fertilizer, is brain-derived neurotrophic factor, BDNF.
Oh, BDNF is critical.
It's heavily featured in the research. But there's a profound nuance here that seems entirely glossed over in normal science communication. The data clearly shows BDNF is not a universal force for growth.
No, not at all.
It exists in two completely antagonistic forms.
The dichotomy of BDNF is a perfect example of how complex these systems are. The molecule is initially synthesized as a precursor protein called pro-BDNF.
And this is the vital nuance, right?
Yes, because pro-BDNF and its mature form, MBDNF, elicit entirely opposing biological responses. When pro-BDNF is secreted, it binds to a receptor called P75.
And what does that do?
Activation of the P75 receptor actively promotes long-term depression. It triggers the retraction of dendritic spines. It can even induce neuronal apoptosis.
Programmed cell death. So the precursor is actively dismantling the network.
It acts as a targeted demolition signal. However, pro-BDNF can be cleaved by extracellular proteases, enzymes that basically snip the protein.
Converting it into mature BDNF.
MBDNF.
Exactly. And this mature form has a high affinity for a completely different receptor, the TRKB receptor. Binding to TRKB initiates the neuroprotective effects.
It promotes the robust LTP, stimulates neurogenesis, and grows new dendrites.
Right. So the adaptability and health of your brain aren't merely determined by how much total BDNF you produce. It's governed by the highly regulated microscopic balance between the demolition signal of pro-BDNF and the growth signal of MBDNF.
That microscopic dance is happening millions of times a second, constantly optimizing the network based on the environment we navigate. But that brings us to a rather dark realization in the literature.
The maladaptive side.
Yeah, because if the brain is this hyper-responsive to environmental inputs, what happens when the environment is objectively hostile, inescapable, chronic?
The data on maladaptive plasticity is deeply unsettling. We often frame neuroplasticity as this inherently positive, self-optimizing feature. But the brain is completely value neutral, isn't it?
Totally value neutral. It simply adapts to survive. Even if an adaptation destroys our cognitive baseline, the clinical data on chronic stress provides the clearest window into this.
Let's talk about the HPA axis.
The hypothalamic pituitary adrenal axis. When you perceive a threat, the hypothalamus releases corticotropin-releasing hormone. That signals the pituitary gland, which signals the adrenal glands to flood your system with glucocorticoids, cortisol.
Which is highly adaptive in the short term. It mobilizes glucose, focuses your attention so you don't get eaten by a tiger.
But the system evolved to handle acute, transient threats, the architecture of modern life.
The incessant digital communication, the constant financial precarity, the terrible sleep.
All of it forces the HPA axis into a state of chronic, unresolved activation.
And the prolonged exposure to those glucocorticoids stops being a performance enhancer and becomes severely neurotoxic.
Highly toxic.
I was looking at the structural imaging data regarding the hippocampus, and it is genuinely haunting. We usually treat major depression or chronic anxiety as purely psychological phenomena, or just invisible chemical imbalances.
But the MRI data shows undeniable structural degradation. The hippocampus is exceptionally vulnerable to glucocorticoid toxicity because it is packed with receptors for these specific hormones.
So under a chronic chemical flood, what actually happens to it?
It undergoes profound morphological changes. We observe severe dendritic atrophy, particularly in the apical dendrites of the CA3 pyramidal neurons.
The branches literally shrink.
They retract and shorten by 20 to 30%. And furthermore, the chronic stress completely halts neurogenesis, the birth of new neurons, in the dentate gyrus.
A 20 to 30% retraction of the receiving branches. The physical volume of the hippocampus shrinks. The studies report up to a 24% volume loss in patients with borderline personality disorder.
And similar, massive reductions in patients with severe treatment-resistant depression.
It perfectly contextualizes the clinical symptoms. The pervasive brain fog, the inability to form new episodic memories, the feeling of being cognitively anchored to a single dark state.
Because the physical hardware required to process new contexts and learn new emotional responses has literally withered away.
But the tragedy of this maladaptive plasticity is compounded when you look at what's happening to the amygdala at the exact same time.
Right, the contrast is stark. While the hippocampus is undergoing severe atrophy, the amygdala, the brain's primary threat detection and fear processing center, is experiencing the exact opposite phenomenon.
Growing.
Under the same chronic glucocorticoid exposure, the neurons in the basolateral complex of the amygdala undergo hyperplasticity. They increase their dendritic arborization, they form new, highly excitable synapses.
So the brain is actively upgrading its threat detection hardware while simultaneously dismantling its logic and memory centers.
It's a devastating structural realignment. The result is a neural architecture that is incredibly efficient at detecting danger, wildly hypervigilant, but completely lacks the hippocampal volume required to contextualize that danger.
It can't realize that a threat has actually passed. It just stays panicked.
Exactly. And the timeline for this kind of physiological rewiring can start alarmingly early. The epidemiological data from the World Health Organization regarding early life stress is staggering.
Yeah, they define early life stress as sustained adverse experiences, profound neglect, extreme poverty, exposure to violence, occurring anywhere from birth to 18 years of age.
And they estimate that 1 billion adolescents are exposed to these conditions globally every year. 1 billion.
The literature shows that exposing a developing brain to that level of sustained HPA axis activation fundamentally alters its developmental trajectory. The regulatory feedback loops become compromised.
They basically get locked in the on position. We see epigenetic changes, specifically DNA methylation, that permanently alter how genes related to stress resilience are expressed.
And structurally, we see decreased gray matter volume and a severely compromised prefrontal cortex.
And the downstream behavioral consequences of that early structural deficit are massive, particularly regarding the propensity for addiction.
Which is a huge theme in this stack.
When the brain's internal regulatory systems are structurally degraded early in life, the organism naturally seeks external, highly potent chemical regulation to stabilize its internal state.
The data indicates that experiencing multiple adverse childhood events exponentially increases the statistical probability of severe substance use disorders and alcohol dependence in adulthood.
And when we look at the cellular environment driving these depressive and addictive behaviors, the research highlights a massive paradigm shift. For decades, neuroscience was almost entirely neurocentric.
Just focused on the neurons.
Right, but the literature here demonstrates that glial cells, specifically astrocytes and microglia, are the hidden architects of this maladaptive state.
This reevaluation of glial cells is incredible. Astrocytes are supposed to provide vital metabolic and structural support to neurons.
But during chronic stress, they actively undergo atrophy in the prefrontal cortex. They retract their processes, and they fail to clear glutamate from the synaptic cleft.
Which contributes to that excitotoxicity we discussed earlier.
Exactly. And simultaneously, microglia, which are the resident immune cells of the central nervous system, shift from a resting, surveying state into a highly aggressive, activated phenotype.
This leads right into the cytokine hypothesis of depression. The data shows these activated microglia just pouring out pro-inflammatory cytokines, specifically tumor necrosis factor alpha, or TNF-alpha.
Creating a localized state of intense neuroinflammation. This inflammation actively suppresses neurogenesis and drives behavioral despair.
It reframes severe depression. It's not just a structural issue or a chemical imbalance. It's essentially an autoimmune-like inflammatory disease of the brain.
It really is. And building on that inflammatory model, we can bridge directly into how addictive substances and really any unnatural dopamine spikes weaponize these exact mechanisms.
Let's dive deep into this. The hijacked reward system. Why does our brain chase dopamine so strongly? Because the dopamine research here, specifically regarding modern digital addiction and substance abuse, is mind-blowing.
Well, to answer why we chase it, you have to look at evolution. The literature on the mesolimbic pathway, specifically the dopaminergic projections from the ventral tegmental area to the nucleus accumbens.
The VTA to the NROC.
Right. This circuitry highlights an evolutionary mismatch. Our brains evolved this intricate reward system to reinforce rare, highly effortful behaviors that were absolutely critical for survival.
Finding calorie-dense food in a famine, navigating to safe shelter, reproduction.
Exactly. Dopamine acts as the ultimate teaching signal for survival.
But the research emphasizes that dopamine isn't just the pleasure chemical that pop culture makes it out to be, right?
Not at all. The classic experiments involving primate learning show that dopamine neurons spike not when a reward is received, but in anticipation of a predicted reward.
It is a molecule of craving, of motivation.
And most importantly, of structural reinforcement. It instructs the NMDA receptors to aggressively hardwire the specific sequence of actions that led to the reward.
There's literally building the physical habit loops.
Yeah.
And that is exactly where the evolutionary mismatch becomes lethal in the modern world.
Lethal is the right word.
Because modern addictive drugs, or even hyper-engineered digital stimuli,
Like infinite social media feeds, TikTok, Instagram.
right, they hijack this exact system. They deliver a highly concentrated, completely unnatural surge of dopamine that completely overwhelms the brain's homeostatic limits.
A massive dopamine flood, and this flood forcefully induces widespread aberrant synaptic adaptations in the nucleus accumbens.
It's rogue plasticity.
Addiction at the cellular level is a form of rogue, drug-evoked neuroplasticity. The brain's survival architecture is basically tricked into prioritizing the acquisition of the drug or the digital hit above all other biological imperatives.
And there's this wildly counterintuitive study in the stack regarding this process. It looks at cocaine sensitization and microglial activation. You would assume the brain's immune system just suffers under the chemical assault.
But the data shows it actually mounts a targeted defense.
A physical defense against the addiction.
Yes. The immune response to cocaine is a fascinating physiological countermeasure. Researchers discovered that repeated cocaine administration robustly activates the striatal microglia.
The same immune cells driving inflammation and depression.
The very same. And just as we saw in the depression models, these activated microglia synthesize and release the inflammatory cytokine TNF-alpha. But in the context of the nucleus accumbens and cocaine,
this localized burst of TNF-alpha actually initiates a protective form of long-term depression.
it actively depresses and weakens the glutamatergic synaptic strength. It is effectively trying to limit the development of behavioral sensitization to the drug.
The brain's immune system recognizes the runaway rogue plasticity as a pathogen. It actively releases inflammatory agents to degrade the structural highways the cocaine is trying to build. That is incredible.
And the twist in the study regarding abstinence completely reframes our understanding of relapse and craving.
What happens when they stop?
The researchers found that after a period of prolonged abstinence, the microglia return to a morphologically resting state, but they remain biochemically primed.
Like they remember the addiction.
Exactly. And if the subject is then exposed to a very mild, non-specific immune trigger.
They used a TLR4 agonist in the study, right? Which
Yeah.
just simulates a mild bacterial infection.
Right. When exposed to that mild immune trigger, these primed microglia rapidly reactivate. They dump TNF-alpha back into the striatum, depress the synaptic strength again, and successfully suppress cocaine-seeking behaviors long after the drug use has stopped.
So a mild peripheral immune response could theoretically suppress neurological craving because the microglia possess an immunological memory of the addiction.
It's a massive leap forward in how we view control and reset mechanisms in the brain.
And speaking of massive leaps, the data on the lateral habenula fundamentally alters how we view the physical roots of anhedonia. If the VTA is the engine of motivation and dopamine,
The lateral habenula is the ultimate emergency brake.
it's this small epithalamic structure that has become central to the modern understanding of major depressive disorder, the brain's anti-reward center.
When an organism anticipates a reward and the reward is withheld, or when the organism experiences a highly aversive, punishing stimulus, the lateral habenula fires intensely.
And its projections travel down to the VTA and the raphe nuclei, actively suppressing the release of both dopamine and serotonin.
The structural data here is profound. In clinical models of severe depression, the lateral habenula exhibits massive hyperactive plasticity,
The excitatory synapses are strengthened.
and the region becomes chronically overactive. It is continuously firing, keeping the brakes permanently clamped down on the brain's motivational and mood-regulating centers.
So the inability to feel pleasure, that crushing weight of anhedonia, isn't just a passive absence of serotonin or dopamine.
It is an active, aggressive suppression driven by a hyperplastic anti-reward center.
And the specific cellular mechanism driving this hyperactivity brings us right back to the glia.
It does. The literature highlights the role of astroglial KIR 4.1 channels in the lateral habenula.
These inward rectifying potassium channels expressed exclusively on the astrocytes surrounding the neurons.
In depressive states, these channels become severely upregulated. This massive increase in potassium buffering completely destabilizes the extracellular environment.
It forces the nearby neurons into an abnormal, rapid burst-firing pattern.
The astrocytes are effectively short-circuiting the neurons, driving the very hyper-excitability that shuts down the reward system.
Okay, so let's summarize where we are. We are looking at a system that is physically degraded. We have a shrunken hippocampus, an overgrown, hypervigilant amygdala, inflamed microglia, a hijacked dopamine system driving rogue plasticity, and an anti-reward center locked in a state of rapid-fire suppression by its own support cells.
It paints a grim picture.
It really does. So the critical question transitioning into the clinical data is, how do we force this deeply entrenched hostile architecture to dismantle itself and rebuild? How do we rewire for recovery?
Well, the traditional pharmacological approach has always relied on the slow, agonizing process of gradual neurogenesis. Standard SSRIs like fluoxetine or Prozac,
Why do they take weeks or months to work?
because they operate by inhibiting the reuptake of serotonin, which instantly floods the synapse with the neurotransmitter. But the clinical efficacy lags because the serotonin itself is not the cure.
It's merely the first slow signal.
Right. It's the start of a massive downstream cascade that eventually leads to an upregulation of mature BDNF and the slow, arduous birth of new neurons in the dentate gyrus.
It's a grueling timeline when a patient is in acute crisis.
Exactly. But the literature on psychoplastogens, these compounds capable of inducing rapid structural neuroplasticity, represents a true paradigm shift.
The data on subanesthetic infusions of ketamine, it is nothing short of revolutionary. We're talking about reversing severe structural atrophy not in months, but in hours.
The mechanism of action for ketamine is a masterclass in exploiting the brain's complex circuitry. Ketamine is an NMDA receptor antagonist.
It blocks the very receptor we said was crucial for learning.
But at low clinical doses, it exhibits a profound preference. It selectively blocks the NMDA receptors located specifically on inhibitory GABAergic interneurons in the prefrontal cortex.
And these interneurons normally act as localized brakes, constantly suppressing the activity of the surrounding excitatory pyramidal neurons.
So by selectively blocking the inhibitor, ketamine essentially cuts the brake lines.
The prefrontal cortex experiences a massive, immediate surge of excitatory glutamate.
Precisely. This sudden, robust glutamate surge bypasses the blocked NMDA receptors and violently activates AMPA receptors.
And what does that intense AMPA activation do?
It triggers the release of mature BDNF and initiates a profound intracellular signaling cascade involving the mammalian target of rapamycin. The mTOR pathway.
Activation of MPOR is the biological equivalent of throwing a massive switch for cellular construction.
It immediately begins the synthesis of new synaptic proteins. Researchers utilizing advanced two-photon microscopy have observed the physical growth of new functional dendritic spines in the prefrontal cortex within mere hours of administration.
It rapidly rebuilds the exact synaptic connections that chronic stress and toxic cortisol levels destroyed. The idea of medically induced hyperaccelerated structural remodeling is incredible.
It is.
But the literature pushes this even further when discussing the concept of critical periods. Here's where it gets really interesting. We all recognize that early childhood is a time of terrifyingly efficient neuroplasticity.
Kids effortlessly wire complex motor patterns. They acquire entirely new phonemic structures for language like it's nothing.
But as adults, that level of fluid adaptability seems entirely lost.
Because the brain actively restricts massive structural plasticity as we age. It has to preserve essential functional stability.
Once the foundational circuits of the visual, auditory, and motor systems are established, the brain deploys these potent molecular brakes.
It downregulates certain growth factors and literally wraps mature neurons in dense extracellular matrix structures called paraneuronal nets.
Paraneuronal nets. They physically lock the synapses in place, severely limiting further structural changes. The critical period closes and the architecture solidifies.
But the data suggests we can artificially dissolve that solid architecture.
The visual cortex study involving fluoxetine.
Right. Researchers were able to reopen ocular dominance plasticity in fully mature adult subjects. Their visual systems reverted to a state of hyperadaptability comparable to a developing child.
The fluoxetine study proved that the brakes are not permanent. They are actively maintained and can be pharmacologically disrupted.
However, that specific intervention was highly transient. The plasticity window remained open only as long as the drug and its active metabolites were present in the cerebrospinal fluid.
Once cleared, typically within 7 days, the paraneuronal nets restabilized and the critical period slammed shut again.
Which sets the stage for the most astonishing proof of concept trial in this entire stack.
The Valproate Absolute Pitch Study. Absolute pitch, the ability to identify or recreate a musical note without a reference tone, is the textbook example of a trait bound by a strict critical period.
The consensus has always been that if you don't receive targeted musical training before the age of 7, the auditory cortex loses the capacity to wire that specific skill.
It was considered a biological impossibility for an adult.
Until the application of Valproate. Valproate is traditionally utilized as an anticonvulsant and mood stabilizer. But fundamentally, it operates as a potent epigenetic modulator.
Specifically, it acts as a histone deacetylase inhibitor.
Inside the cell nucleus, DNA is tightly wound around structural proteins called histones. This tight spooling prevents the cellular machinery from accessing and reading specific genes.
By inhibiting histone decetylation, Valproate forces the chromatin to uncoil.
It exposes vast arrays of genes, specifically those related to profound structural neuroplasticity that had been silenced since childhood.
And the methodology of this study was incredibly precise. They recruited adult men with absolutely no absolute pitch ability.
They titrated the Valproate to maintain a highly specific serum concentration between 350 and 700 micromoles per liter, ensuring the epigenetic modulation was active without inducing toxicity.
And while maintaining that specific blood concentration, they subjected the men to an intensive 2-week absolute pitch training protocol.
The results shattered previous neurological assumptions. The cohort receiving the Valproate performed significantly better than the placebo group. They successfully acquired foundational aspects of absolute pitch.
The drugs systematically degraded the molecular brakes, transiently returning the adult auditory cortex to a juvenile, highly plastic state. They chemically rolled back the developmental clock.
To allow a mature brain to absorb a skill that was supposedly lost to time, it's a breathtaking proof of concept.
It really is. But the literature isn't solely focused on pharmacology. The advancements in non-invasive, targeted physical interventions are producing remission rates that rival the most potent chemical treatments.
The data on transcranial magnetic stimulation, particularly the Stanford Saint protocol, it completely redefines psychiatric intervention.
TMS operates on the principle of electromagnetic induction. By placing a specialized magnetic coil over the scalp, clinicians can deliver highly focused magnetic pulses that penetrate the skull.
And induce highly specific, localized electrical currents in targeted cortical regions. By precisely modulating the frequency and pattern of these pulses, they can force a specific brain network to undergo either LTP or LTD.
And What the team at Stanford did with the same protocol was hyper-optimize this process. They didn't just guess where to aim the magnet.
No, they utilized accelerated functional MRI to map the unique neural topography of each individual patient.
They specifically located the exact coordinates in the dorsolateral prefrontal cortex that exhibited the strongest negative functional connectivity to the subgenual anterior cingulate cortex.
A region deeply implicated in the pathology of severe depression. It is the absolute pinnacle of personalized precision neuroengineering.
Once they mapped that exact target, they delivered a highly accelerated, incredibly dense schedule of intermittent theta burst stimulation.
They compressed months' worth of standard TMS treatment into just 5 intensive days.
And the clinical outcomes were unprecedented. In a cohort of patients suffering from severe treatment-resistant depression, individuals who had failed multiple pharmacological trials,
Nearly 80% achieved full clinical remission within days.
they structurally rebuilt the regulatory networks of the brain using nothing but focused magnetic fields.
And we are seeing the same ethos of precision engineering applied to neurodegenerative disorders. The NIH Brain Initiative data highlights the leap from traditional deep brain stimulation to adaptive deep brain stimulation, or ADBS, for Parkinson's disease.
Traditional DBS is kind of a blunt instrument. It involves surgically implanting electrodes into specific basal ganglia targets and delivering a constant, unvarying electrical pulse.
To disrupt the pathological firing patterns that cause tremors and rigidity.
But adaptive DBS incorporates advanced closed-loop technology.
The implanted system continuously records the patient's localized field potentials, the real-time electrical chatter of the brain.
It utilizes highly sophisticated algorithms to instantly analyze those signals and dynamically adjust the amplitude and frequency of its stimulation.
Millisecond by millisecond, delivering exactly the amount of current required precisely when the brain needs it.
It is a fully autonomous, intelligent pacemaker for the brain, seamlessly integrating with the patient's shifting neurophysiology. The trial data shows it significantly outperformed traditional DBS, reducing the most distressing motor symptoms by an additional 50%, while dramatically lowering the energy output and side effects.
It's incredible.
But we have to bring this incredible high-level science back down to the reality of the listener. How do we hack our own plasticity?
Right, because we cannot all access fMRI-guided magnetic helmets, closed-loop brain implants, or sub-anesthetic ketamine infusions this week.
The critical transition here is looking at the literature regarding endogenous plasticity. The data clearly demonstrates that our daily mundane behaviors exert a massive structural influence on our neural architecture.
If we look at the research on physical exercise, it's essentially described as an endogenous psychoplastogen.
The ultimate hack. The clinical framing of exercise must shift from pure cardiovascular or metabolic maintenance to active structural brain remodeling.
When an individual engages in acute high-intensity aerobic exercise, they initiate a systemic, multi-organ chemical cascade that directly targets the central nervous system.
The data reveals that a single session of vigorous aerobic output can induce a 32 to 45% surge in circulating levels of mature BDNF.
A 45% increase in the brain's primary growth factor, generated entirely by physiological exertion.
And the mechanism connecting the peripheral body to the brain during exercise is deeply fascinating, specifically the liver-brain axis.
The liver plays a surprisingly critical role in neuroplasticity. During intense, sustained physical exertion, hepatic glycogen stores are depleted, and the liver shifts its metabolic profile to begin oxidizing fatty acids.
This process results in the robust production of ketone bodies, specifically beta-hydroxybutyrate.
This molecule is highly soluble, rapidly enters the systemic circulation, and easily crosses the blood-brain barrier.
And once it enters the brain, particularly the hippocampus, it doesn't just act as an alternative fuel source. The data shows it acts as a highly potent epigenetic regulator.
Exactly. Beta-hydroxybutyrate functions as an endogenous inhibitor of histone deacetylases, very similar in mechanism to the valproate we discussed earlier.
So by inhibiting these enzymes in the hippocampus, beta-hydroxybutyrate forces the chromatin surrounding the BDNF gene to unspool and remain in an open, transcriptionally active state.
The physical act of running forces the liver to send a direct molecular signal to the hippocampus, commanding it to unlock its DNA and significantly upregulate the production of BDNF,
thereby accelerating synaptic plasticity and neurogenesis. And the profound structural impact of exercise scales up from the microscopic epigenetic level to the macroscopic network level.
The neuroimaging data shows that sustained aerobic exercise fundamentally alters the functional connectivity between the brain's large-scale intrinsic networks, specifically the default mode network and the central executive network.
The default mode network, the DMN, is primarily active during internally directed cognition, daydreaming, autobiographical memory retrieval, and crucially, rumination.
In depressive and highly anxious states, the DMN becomes hyperconnected and dominant. The individual becomes trapped in an endless loop of negative self-referential thought.
The central executive network, the CEN, conversely governs externally directed attention, complex problem solving, and cognitive control.
The literature demonstrates that regular exercise strengthens the white matter tracks, the physical infrastructure connecting these networks.
It enhances the brain's structural capacity to willfully disengage from the ruminative trap of the default mode network and fluidly transition into the focused, engaged state of the central executive network.
You are physically building the roads required to steer your own attention.
And if BDNF is the cellular construction crew building these roads, the nutritional data dictates the quality of the raw materials they are using.
The literature on the gut-brain axis and specific macronutrients reveals that you cannot sustain high-level neuroplasticity on a structurally deficient diet.
The lipid composition of the brain is incredibly specific, and it relies heavily on dietary inputs. Omega-3 polyunsaturated fatty acids, specifically docosahexaenoic acid, or DHA, are absolutely foundational.
Because the neuronal cell membrane is not a static wall. It is a highly fluid, dynamic lipid bilayer.
The receptors we spend so much time discussing, the NMDA and AMPA receptors, must be able to move, cluster, and embed themselves within this membrane to execute long-term potentiation.
DHA provides the essential biochemical fluidity required for this movement. A diet deficient in DHA results in rigid, inflexible neuronal membranes, fundamentally impairing the physical mechanics of synaptic plasticity.
The data on polyphenols is equally critical, particularly compounds like curcumin found in turmeric. The literature shows that curcumin doesn't just offer mild antioxidant support.
It actively penetrates the blood-brain barrier and engages specific molecular pathways. Curcumin has been shown to robustly activate the AKT-CRB signaling pathway.
CRB is a highly critical transcription factor that, once activated, binds directly to the DNA to massively upregulate the transcription of BDNF.
Furthermore, curcumin exerts profound anti-inflammatory effects within the central nervous system, directly antagonizing the microglial activation and TNF-alpha production we previously identified as the primary drivers of depressive pathology.
And the gastrointestinal tract itself is actively modulating the brain's chemical environment. The gut microbiome is responsible for fermenting dietary fiber into short-chain fatty acids, like butyrate.
These SCFAs interact directly with the enteric nervous system and transmit potent regulatory signals straight up the vagus nerve to the brain, suppressing neuroinflammation and modulating the synthesis of key neurotransmitters.
Which brings us to the most universally consumed psychoactive compound globally, caffeine.
Ah, yes.
The data on how caffeine and its primary metabolites theobromine, paraxanthine, and theophylline interact with the brain's plasticity mechanisms is remarkably nuanced.
Caffeine operates fundamentally as an antagonist to adenosine receptors. As neurons fire and consume ATP for energy throughout the waking day, adenosine accumulates in the extracellular space.
It binds to its receptors, slowly hyperpolarizing the neurons and exerting a profound sleep pressure.
Caffeine's primary mechanism of promoting wakefulness is by competitively blocking the adenosine A1 receptors, which instantly removes this inhibitory brake and triggers a widespread release of excitatory neurotransmitters.
But the literature highlights that its role in structural plasticity is driven by its interaction with an entirely different receptor subtype.
The crucial mechanism for our discussion is its blockade of the adenosine A2A receptors. These receptors are heavily concentrated in the striatum and the hippocampus.
The data indicates that antagonizing the A2A receptors physically lowers the threshold required to induce long-term potentiation.
By blocking this specific receptor, caffeine actively sensitizes the synapses, enhancing structural adaptability and significantly boosting spatial memory consolidation and overall cognitive flexibility.
It is actively lowering the molecular barrier to entry for network construction. But the ultimate prerequisite for all this construction,
the absolute non-negotiable requirement for neuroplasticity,
is sleep. We can flood the brain with BDNF, optimize our DHA levels, and drink all the coffee we want, but the physical reality is that the actual structural remodeling does not occur while we are awake.
Wakefulness is the period of acquisition. We encounter stimuli, activate the NMDA receptors and initiate the calcium cascades.
But the waking brain is metabolically constrained. It cannot simultaneously process complex environmental inputs and undergo massive structural reorganization.
Sleep is the state of active, aggressive consolidation. During the deepest stages of non-REM sleep, the brain utilizes highly synchronized electrical oscillations.
Specifically, sleep spindles and sharp wave ripples originating in the hippocampus.
It uses those to systematically replay the exact neural firing patterns that occurred during the day.
It replays the day's code, but at a vastly accelerated rate, forcefully cementing the long-term potentiation pathways that represent vital new information.
While simultaneously utilizing long-term depression to widely scale down and prune the synapses that simply represent background noise.
It is the ultimate expression of synaptic homeostasis. If an individual suffers from chronic sleep deprivation, they force their brain into a state of aberrant, chaotic cortical excitability.
The synapses remain saturated from the previous day's inputs. Without the localized pruning and structural cementing that occurs exclusively during deep sleep, the brain is physically incapable of inducing new functional LTP the following morning.
The capacity for learning, emotional regulation, and psychological resilience is structurally compromised.
Totally compromised.
We have mapped an immense and intricate landscape today. We started at the fundamental, microscopic architecture of change, examining the precise voltage requirements of the NMDA receptor and the delicate high-stakes balance between the demolition signals of pro-BDNF and the vital growth signals of mature BDNF.
We navigated the devastating structural reality of maladaptive plasticity. We saw how the chronic, toxic flood of cortisol physically retracts the dendritic branches of the hippocampus, driving memory loss and cognitive rigidity,
while simultaneously forcing the amygdala into a state of hypervigilant panic. We examine how profound early life stress epigenetically degrades the brain's regulatory systems, setting a dangerous trajectory for future addiction.
We explored the intricate mechanisms of the mesolimbic pathway, observing how the evolutionary imperative of dopamine is hijacked by supernormal digital and chemical stimuli, inducing runaway, drug-evoked structural changes.
We witnessed the brain's immune system desperately deploying localized inflammation to fight off this rogue rewiring, and we uncovered the cellular pathology of despair, how hyperactive astrocytes in the lateral habenula actively short-circuit the brain's motivational core.
We also documented the breathtaking frontiers of neurological intervention. We analyzed the rapid mTOR-driven structural regrowth triggered by psychoplastogens like ketamine.
We saw the precision engineering of fMRI-guided magnetic stimulation pulling patients out of intractable depression in days, and adaptive closed-loop implants seamlessly pacemaking the compromised circuitry of Parkinson's patients.
We explored the astonishing reality that epigenetic modulators like valproate possess the sheer molecular force to dissolve perineuronal nets and roll back the developmental clock, briefly returning the rigid adult mind to the hyperplastic state of infancy.
And finally, we grounded this vast scientific revolution in the undeniable structural power of endogenous plasticity. The realization that an intense aerobic effort forces the liver to chemically command the brain to unlock its DNA and synthesize growth factors.
The knowledge that the structural integrity of our memories relies entirely on the precise lipids we consume and the sanctity of our deep sleep architecture.
The ultimate synthesis of this literature is a mandate of absolute agency. Every specific environment you immerse yourself in, every agonizing physiological effort you endure, every specialized macronutrient you ingest, and every hour of deep consolidation you protect is an active physical instruction to your biological hardware.
You are not a passive observer of a static machine. You are the active architect of a continuously evolving neural landscape. Understanding your brain, your dopamine, your stress, it's the first step to taking control of your attention and your biology.
It is the most empowering realization in modern biology.
It really is. And as we conclude this deep dive, I want to linger on the profound implications of that valproate proof of concept trial. We possess clinical evidence that it is biochemically possible to temporarily dissolve the structural breaks of adulthood and reopen the critical learning windows of childhood, allowing mature individuals to rapidly acquire a neurological trait previously deemed biologically impossible.
It fundamentally destabilizes our concept of fixed adult identity.
It raises an incredibly provocative question regarding the future of human optimization. If the rapid progression of this neuroscience eventually perfects the ability to selectively and safely unlock these highly plastic critical periods on demand, What foundational human traits, what impossibly complex foreign languages, or what entirely novel, high-dimensional ways of perceiving reality might you choose to learn from absolute scratch? If the architecture of the adult mind becomes completely, entirely malleable by design, who would you actively choose to become? Welcome back to the deep dive. We're really thrilled you're joining us today.
Yeah, it's great to be here.
You know, looking at the listeners who usually tune into this show, you're the kind of person who doesn't just want the surface level summary.
Right. You want the actual mechanics.
Exactly. You are a lifelong learner. You want to know how things actually work. And if you've ever been curious about habit formation or how behavioral loops are broken.
Or what exactly is happening under the hood of your own mind when you try to change a deeply ingrained routine.
Yeah, today's focus is going to be incredibly illuminating.
It is a fascinating area of study because we're zooming in on a very precise, very critical, and honestly, often misunderstood window of neurological time.
We really are. Our mission today is to unpack the exact neurological timeline of quitting cannabis.
But we aren't doing a broad, vague overview of, like, getting clean.
No.
Yeah.
We are putting a highly focused magnifying glass over a specific window, days 7 to 14 of abstinence.
Week 2.
Week 2. And to do this, we've pulled a massive stack of clinical literature.
Oh yeah, we've got some heavy hitters today.
We really do. We're going to be dissecting high-resolution PET scan imaging studies, comprehensive neurobiological reviews on the endocannabinoid system, and precise behavioral research on withdrawal timelines.
And the reason we're targeting this second week of abstinence is that when you look at the clinical data, this 7 to 14-day window represents a profound biological turning point.
It's a huge shift.
It is. It's a period of intense structural and chemical reconstruction in the brain that frequently gets overshadowed by the acute physical symptoms of the first few days.
Right, the sweating and the shaking. But before we get to week 2, since you're already familiar with standard forward neurotransmission, the basic process of a neuron firing, releasing a chemical, and that chemical crossing the synapse.
To bind to the next neuron, right?
Exactly. Let's jump straight into the anomaly that is the endocannabinoid system. To understand what the brain is recovering from in week 2, we have to establish the baseline of the hack.
Right. The endocannabinoid system essentially operates in reverse.
Which is wild to think about.
It uses what we call retrograde signaling. So imagine a receiving neuron is being bombarded by way too much excitatory input, like an influx of glutamate.
It's getting overwhelmed.
Right. It needs a way to tell the sending neuron to back off. So to do this, the receiving neuron synthesizes its own natural cannabinoids on demand.
Like it just makes them right then and there.
Exactly. Specifically molecules like 2-arachidinoyl glycerol or 2-AG and anandamide.
And instead of firing forward, those molecules travel backward across the synaptic cleft.
Precisely. They travel backward and bind to cannabinoid type 1 receptors, CB1 receptors, located on the presynaptic terminal.
The sending neuron.
Right. And when 2-AG binds to that CB1 receptor, it acts as a braking mechanism. It physically inhibits the release of further neurotransmitters, whether that's excitatory glutamate or inhibitory GABA.
So it's like the brain's master volume control.
That's a great way to put it. It maintains cellular homeostasis.
Okay, so when someone consumes cannabis, THC enters this really delicate, highly calibrated feedback loop. I know THC acts as a structural mimic of these natural cannabinoids, but is it just a stronger version of 2-AG, or does it fundamentally operate differently?
It fundamentally operates differently in terms of its affinity and its persistence.
How so?
Well, 2-AG and anandamide are fragile. They're synthesized on demand, they do their job, and then they're rapidly broken down by enzymes like FAAH and EVL.
They don't stick around.
No, they don't. THC, on the other hand, is a highly potent exogenous compound that is not easily broken down by those local enzymes.
Right.
It floods the system and binds persistently to those CB1 receptors. It doesn't just turn the volume knob down. It rips the knob right off.
Wow, rips the knob off. And the downstream effect of this persistent binding is massive, right? Especially concerning dopamine.
Yes, through a process called GABAergic disinhibition.
Okay, break that down for us.
So by persistently binding the CB1 receptors on GABA neurons, THC stops the release of GABA. And since GABA is the chemical that normally puts the brakes on dopamine release,
Removing those brakes causes artificial surges of dopamine.
exactly, surges in the brain's reward centers, like the nucleus accumbens.
But the brain is highly adaptive. It doesn't just, passively accept this persistent overstimulation. It initiates defensive countermeasures.
It initiates a profound structural defense.
Yeah.
To survive the constant overstimulation from THC, the brain downregulates.
Downregulates, meaning it literally internalizes its CB1 receptors.
Right. It pulls them off the surface of the neurons and dismantles them so they can no longer be activated. It drastically reduces receptor
To
density.
protect itself.
Yes. And simultaneously, because the THC is artificially driving dopamine release, the brain suppresses its own natural synthesis of dopamine.
Which sets the perfect stage for what happens when you quit.
It does.
Because if you stop consuming cannabis, those first few days, days 1 to 3, are famously difficult. The clinical literature frames this through the opponent process theory.
Right.
The exogenous THC is suddenly gone, but all those heavy-handed counter-regulatory mechanisms,
The missing receptors, the suppressed dopamine,
yeah, they're still active. They are running unopposed.
and that biological deficit translates directly into the acute withdrawal syndrome. Sweating, intense irritability, headaches, profound anxiety, and just complete sleeplessness.
Your central nervous system is essentially in a state of unregulated shock.
It is.
But let's say you push through that. You survive the opponent process shock of the first 72 hours. You cross the threshold into week 2, days 7 to 14. This brings us to the first major biological event in our targeted timeline,
Brain receptors start resetting.
the rebound.
This is where the true neuroadaptive reversal begins. With the persistent binding of THC removed, the brain recognizes that it's operating with a severe deficit of CB1 receptors.
It realizes it doesn't need the defensive armor anymore.
Exactly. And it begins the process of upregulation.
I really want to spend some time on the exact data we pulled for this because the methodology is just fascinating. We're looking at a high-resolution research tomography PE scan study.
An HRRT scan.
Right, using a specific radioactive tracer called OMAR. Well, 11-CoMAR. What exactly does this technology allow researchers to see?
HRRTP is an incredibly sophisticated imaging technique. The OMAR radio tracer is an isotope designed to specifically seek out and bind to CB1 receptors in the brain.
Okay.
And when it binds, it emits positrons that the scanner detects, giving us a highly detailed three-dimensional map of exactly where and how dense these receptors are across the cortex.
So they can literally see the receptors.
Yes.
The researchers took a group of chronic daily cannabis users and scanned their brains at baseline, meaning they weren't intoxicated during the scan, but they were still active users who hadn't begun the quitting process yet.
Right, and they compared this map to a control group of healthy non-users.
And the baseline deficit is striking. The chronic users showed a 15% lower CB1 receptor availability across almost all cortical brain regions.
And we really need to emphasize the statistical weight of that finding. In the study, this 15% reduction corresponded to a Cohen's d effect size of -1.11.
Now, a 15% reduction sounds significant on paper, but the brain is incredibly plastic, right? It's resilient. Does a 15% drop in receptor density actually correlate linearly to a noticeable loss of cognitive function? Or is the brain compensating in other ways before the person even decides to quit?
That is an excellent question. The brain absolutely attempts to compensate by shifting loads to other neural networks,
Yeah.
which is why chronic users don't completely lose their ability to function.
They're still going to work. They're still living.
Exactly. However, the endocannabinoid system is so ubiquitous and so central to modulating other neurotransmitters that a -1.1 effect size just cannot be fully compensated for.
It's too big of a hit.
Right. For context, in behavioral sciences, a Cohen's d of 0.8 is considered a large effect. So a -1.11 represents a profound structural alteration, one that directly correlates to the cognitive dulling, the memory deficits, and that pervasive brain fog that chronic users report.
Because the volume control knobs for the entire cortex are literally missing.
Exactly.
But the most crucial data point from this OMAR study isn't the baseline damage, it's the timeline of the recovery.
Yes.
They monitored these individuals as they were placed in an inpatient setting and required to abstain.
And the speed of the neuroplasticity is staggering.
Truly.
After just 2 days of monitored abstinence, the scans showed that significant upregulation had already begun.
Just 2 days.
The researchers actually noted a robust negative correlation between the severity of the acute withdrawal symptoms on day 2 and the availability of these newly upregulating receptors.
So as the individuals pushed past the acute phase and entered our focus window days 7 to 14, the scans showed this receptor recovery was well underway.
The receptors were actively migrating back to the surface of the cell membranes.
And the brain was restarting the production of its own natural cannabinoids.
Which is the exact biological mechanism that explains why the intense physical withdrawal symptoms of the first few days start to resolve.
Your body is no longer in a state of complete receptor starvation.
Right. The internal volume controls are being reinstalled. And just to look at the long-term outcome of this study, by 28 days of sustained abstinence, the cortical receptor density in those chronic users was practically indistinguishable from the non-users.
The people who had never touched the drug.
Yes.
That is remarkable. It's almost like stepping out of a blindingly bright room.
Oh, I like that.
Yeah, if the active THC exposure is the blinding light, your brain protects you by forcing you to squint and contracting your pupils. That's the downregulation of the CB1 receptors.
Right.
When you suddenly step out of that room into natural light, which is the act of quitting, you are effectively blind for a moment.
Because your pupils are still pinned.
Exactly. The natural light isn't enough to see by. That's the intense, disorienting withdrawal of days 1 to 3.
That analogy maps perfectly to the neurobiology.
And as you move through week 2, your eyes are finally adjusting, your pupils dilate again, you're upregulating. The natural light of your own internal neurochemistry is finally enough to illuminate the room.
And the direct, noticeable effect of this specific biological reset for the person experiencing it is the lifting of the brain fog.
They report feeling sharper, clearer, and more present.
Entirely because the cortical networks are regaining their natural modulatory systems.
But if the receptors are finally unclogging and the cognitive fog is beginning to lift, it begs an important question. Why do individuals in the 7 to 14-day window still report feeling so profoundly unmotivated, flat, and joyless?
Right, which is the perfect pivot to the second major event of week 2. The dopamine system stabilizes.
We are moving from cognitive clarity to the return of motivation.
To understand this, we have to look at another piece of imaging data we pulled, the 18F-DOPA-PI scan study.
So while the previous scan looked at CB1 receptors, this radiotracer is designed to measure dopamine synthesis capacity.
Specifically in the striatum, a key component of the brain's reward and motor circuitry.
And it's crucial to clarify what synthesis capacity means here. It's not just taking a snapshot of how much dopamine is floating around in that exact second, right?
No, it's measuring the brain's mechanical capability to manufacture dopamine from its precursor, LDOPA.
It's measuring the integrity of the dopamine factory itself.
Exactly. And what the scans show is that chronic cannabis use leads to a severe hypodopaminergic state.
Hypodopaminergic, low dopamine.
Right. Over time, the constant artificial flooding of dopamine caused by THC forces the brain to shut down its own production lines.
Yeah.
The striatum's capacity to synthesize dopamine becomes severely blunted.
Which manifests behaviorally as an emotivational state, or clinical anhedonia, the inability to feel pleasure.
Yes. In the first week of quitting, without the exogenous drug there to force a dopamine release, and with your natural factories effectively shut down, your baseline dopamine levels plummet.
And dopamine is the currency of action. Without it, the brain simply cannot assign value or reward to everyday tasks.
It just can't.
Let's ground this in a real-world adult example. Think about a mundane task like folding a basket of laundry. Or, opening a slightly annoying email from your boss.
Right.
For a healthy brain, these tasks aren't thrilling, but you have enough baseline dopamine to initiate the action, and a tiny microburst of dopamine when you finish it, which registers as satisfaction.
A sense of completion.
Yeah. But in the hypodopaminergic state of week 1, the thought of folding that laundry feels like contemplating climbing Mount Everest.
There is zero chemical incentive to move.
Socializing feels like an exhausting performance. Food tastes like cardboard.
Because the threshold for reward has been set impossibly high by the drug, and your natural chemical reserves are at rock bottom.
It's a brutal combination.
It is. However, during the 7 to 14-day window, a critical shift occurs.
This is the turning point.
Right. The brain registers the prolonged absence of the artificial dopamine floods. It recognizes the severe biochemical drought, and it slowly begins to reopen the natural dopamine factories.
The synthesis capacity begins to normalize.
The dopamine signaling starts to find a fragile balance.
It's a delicate process, isn't it?
It's incredibly delicate. And the clinical community takes this hypodopaminergic state very seriously. In the literature, we see researchers exploring interventions specifically for this.
Right, investigating what are called pro-dopamine regulators.
Yes, there is a complex known as KB220Z that is being studied for its potential to help induce dopamine homeostasis in individuals whose reward circuitry is profoundly disrupted by long-term substance use.
Which really underscores that overcoming this phase isn't just a matter of psyching yourself up.
No, it is a matter of restoring fundamental chemical balance.
So if you are moving through week 2, this biological stabilization is why, seemingly out of nowhere around day 10 or 12, the absolute grayness of the world starts to crack.
Yeah,
You might actually find yourself wanting to go for a walk outside.
you might hear a song on the radio and feel a genuine, spontaneous rush of enjoyment.
Or you might text a friend back because the idea of a conversation suddenly sounds appealing rather than draining.
Your brain's reward thresholds for non-drug reinforcers are dropping back to a healthy baseline. The drug is no longer the only thing that can clear the bar for pleasure.
Natural motivation returns because your striatum is physically synthesizing and releasing dopamine more efficiently in response to normal everyday stimuli.
It is an incredible mechanical shift in how you perceive the value of the world around you.
It really is. But that's what's happening while you're awake. We need to look at the other third of your life.
Ah, yes.
The third major event of days 7 to 14, sleep starts rebuilding. The dream rebound.
Sleep disruption is consistently cited as one of the primary reasons people relapse during the early stages of quitting,
It's so tough.
because THC fundamentally alters sleep architecture. When we talk about sleep, we divide it broadly into non-rapid eye movement, or NREM sleep, which has several stages including deep slow-wave sleep.
And rapid eye movement, or REM sleep.
Right.
Now, REM sleep is the highly active phase of the sleep cycle where we experience the vast majority of our vivid dreaming.
And it is absolutely critical for emotional processing, memory consolidation, and clearing out the psychological debris of the day.
And THC is a potent suppressor of REM sleep.
Very potent. When someone consumes cannabis daily, especially close to bedtime, they are actively truncating their time spent in the REM stage. Over months or years, this creates a massive, unfulfilled biological need.
But the brain doesn't just write off that missing REM sleep.
No, it does not. The brain is meticulous about its requirements. When you stop using THC, the suppression is lifted, and the brain immediately attempts to reclaim the missing REM sleep.
Leading to a phenomenon known as the REM rebound.
Exactly.
The first few days make that impossible. If we look at days 1 to 7, the acute withdrawal phase, users often report severe, crushing insomnia.
They might only manage 2 to 4 hours of broken, restless sleep a night.
So what is happening mechanically there that prevents the rebound from starting immediately?
The acute insomnia is driven by a massive neurochemical stress response. Specifically, the withdrawal triggers spikes in corticotropin-releasing factor, or CRF.
CRF is a primary stress hormone.
Yes, and it interacts heavily with the amygdala, the brain's fear and emotion center. So your central nervous system is essentially locked in a physiological state of high alert and panic.
Just constantly wired.
Right. Your heart rate variability changes, your core body temperature regulation is off, and the physical agitation makes transitioning into the lower brainwave frequencies required for sleep nearly impossible.
But as you transition into week 2, that sheer wired panic begins to dissipate.
Yes. As we discussed earlier, the receptors are upregulating. And crucially, the balance between GABA, the primary inhibitory calming neurotransmitter, and glutamate, the primary excitatory neurotransmitter, starts to stabilize.
The CRF spikes subside.
The physical, agitated inability to sleep wanes, and you can finally initiate and maintain sleep for longer durations.
And this is where the floodgates open. Because you are finally staying asleep long enough to cycle through the NREM stages, you hit the REM phase, and the rebound hits its stride.
The brain begins rapidly processing a massive backlog of suppressed REM sleep.
Instead of the normal 20 to 25% of the night spent in REM, the brain plunges into extended, dense periods of REM sleep.
The analogy I always come back to is that the brain operates like a strict, uncompromising accounting system.
Oh, I love this.
It keeps a precise ledger of the REM sleep it was denied over the years of chronic use. When you quit, it effectively says, you owe me 500 hours of REM sleep, and we are going to collect that debt right now. With interest.
With interest. And the experiential result of this biological debt collection is intensely vivid dreaming.
Very intense.
People who are in the 7 to 14-day window frequently report that their dreams aren't just vivid, they are cinematic, hyper-realistic, emotionally exhausting, and sometimes deeply bizarre or unsettling.
Right.
Why are they so intense? Is it purely the volume of REM sleep, or is the quality of the REM different?
It's actually a combination of both. The volume and density of REM are significantly higher, meaning the periods of dreaming are longer and more frequent. But additionally, because the brain is processing a backlog of emotional memories and psychological states that were previously suppressed, the content of the dreams can be highly charged. The amygdala is highly active during REM, while the logic centers of the frontal lobe are subdued.
Leading to these surreal, intensely emotional narratives.
Exactly.
What's crucial to point out is that individuals often wake up from these hyper-vivid dreams feeling exhausted, and they misinterpret the intensity as a sign that their brain is permanently damaged.
Or that their sleep is permanently broken.
Right.
But in reality, it is the exact opposite. This REM rebound is a vital, normal biological sign of neural healing. The brain is doing the necessary, albeit intense work of clearing the backlog and restoring normal sleep architecture.
So while the overall duration and quality of sleep are slowly improving during days 7 to 14, the intensity of the dreams is an expected feature.
Not a malfunction.
It's part of the recovery process. That is incredibly reassuring context. The brain is just balancing the books. Okay, let's move forward from sleep to waking cognition and specifically to the front of the brain.
Yes, let's do that.
Event number 4 in our week 2 timeline. Prefrontal cortex activity improves. The executive center wakes up.
The prefrontal cortex, the PFC, is arguably the most advanced and evolutionarily recent part of the human brain.
It's what makes us human, really.
Exactly. It's responsible for high-level executive functions, complex decision-making, future planning, working memory, and impulse control. In neurobiology, we refer to the PFC as the top-down regulatory system.
Right, and it operates in constant tension with the subcortical regions, particularly the basal ganglia and the amygdala, which act as a bottom-up system driven by immediate urges, emotional reactivity, and basic reward seeking.
Yes, but chronic cannabis use, especially when initiated early in life, creates a profound structural and functional imbalance between these two systems,
It heavily favors the bottom-up impulse-driven networks.
while suppressing top-down executive control.
The sources included a fascinating structural MRI study focusing on adolescents that really highlights the physical nature of this imbalance.
Yes, the findings of that MRI study were quite striking. The researchers compared structural brain scans of cannabis-abusing adolescents with healthy, non-using controls. They specifically measured regional gray matter volume.
And what did they find?
They found that the cannabis users showed a significant decrease in volume in the right medial orbital prefrontal cortex, the MOPFC.
And the really critical piece of data from that study was the correlation they found regarding age. The decreased volume in the MOPFC correlated positively with the age of first use.
Meaning the earlier the individual initiated chronic cannabis use during those vital developmental years, the more pronounced the structural volume deficit in the prefrontal cortex.
The drug effectively interrupted the normal maturation of the executive center.
It did.
And this structural difference isn't just an interesting quirk on an MRI scan. It translates directly to measurable behavioral deficits. The researchers quantified this using the Barrett Impulsiveness Scale, or the BIS.
They did. The BIS is a widely used psychometric tool that breaks impulsivity down into different subscales, like motor impulsivity, which is acting without thinking.
And cognitive impulsivity, which is making quick decisions.
Right. And the cannabis users scored significantly higher on the BIS, specifically for non-planning impulsivity.
Let's translate non-planning impulsivity into a real-world scenario.
Okay.
Instead of a simplistic analogy like wanting to eat an entire cake, let's look at adult behavioral protocols. Imagine the brain's operation as a complex software system.
I like this.
The subcortical network is running a default background script that constantly seeks immediate dopamine release. Scrolling social media, grabbing a drink, or in this case, seeking cannabis.
The path of least resistance.
Right. Now, the prefrontal cortex is the admin-level protocol that is supposed to override that default script in real time.
It analyzes the situation and says, executing that script will jeopardize our presentation tomorrow morning. Halt execution.
Exactly. Non-planning impulsivity means that admin-level override protocol is offline. You don't anticipate the future outcome, you just let the default script run.
That is a highly accurate way to frame the interaction. During active chronic use, and heavily throughout the acute withdrawal phase of days 1 to 3, the PFC is functionally suppressed.
Because of the glutamate, right?
Right. THC induces a suppression of glutamate, the brain's primary excitatory neurotransmitter in these frontal regions. This creates what neuroscientists call a hypoglutamatergic state in the prefrontal cortex.
So the admin level protocol isn't just ignoring the default scripts. It doesn't have the electrical power to run in the first place. The brakes on your impulses are physically worn out.
Precisely. You are entirely at the mercy of the bottom-up emotionally reactive systems.
But as we transition into the 7 to 14-day window, a profound neurochemical shift occurs.
The suppression begins to lift.
The hypoglutamatergic state begins to resolve.
The glutamate transmission onto GABAergic interneurons within the PFC starts to function normally again.
This biological awakening of the PFC perfectly explains the subjective shift people experience in week 2. In the first 3 days, you are just surviving. You are reacting purely to the physiological urgency of the withdrawal, the stress hormone spikes, and the physical discomfort.
You have no top-down control.
But as you cross into the second week, you regain the ability to step back.
As the glutamate transmission restores connectivity in the frontal lobe, your focus noticeably improves.
Your working memory expands.
And crucially, your impulse control strengthens. The admin-level override protocol comes back online.
So sticking to your long-term goal of remaining abstinent actually becomes biologically easier because your executive center is reasserting its regulatory power over the reactive, bottom-up systems.
Exactly.
It is empowering to realize that the sudden sense of mental clarity and resolve you feel in week 2 isn't just a random mood swing, or you suddenly finding more willpower.
No, it is the literal physical restoration of your brain's frontal lobe connectivity.
And this restoration perfectly tees up our 5th major point. Cravings begin to decrease. The psychological shift.
To understand this shift, we need to clearly differentiate between the distinct phenotypes of craving.
Okay, let's break that down.
In the first 3 to 7 days, the cravings are intensely physical. That is your central nervous system, deep in the opponent process state, screaming for the exogenous cannabinoids it is grown reliant upon to regulate basic functions like body temperature, digestion, and sleep.
It's a physiological demand. It manifests as sweating, shaking, and an overpowering somatic urge.
But as we move into the days 7 to 14 window, the nature of those cravings fundamentally changes.
The physical intensity dramatically drops.
The cravings become less frequent, and when they do occur, they present more as intrusive, fleeting thoughts rather than overpowering bodily demands.
Let's contextualize this shift using the clinical framework developed by Koob and Volkow.
Oh, yes.
They describe addiction and withdrawal in stages, and week 2 aligns heavily with the preoccupation anticipation stage.
Yes. In the earlier stages of addiction, the brain assigns massive artificial importance to the drug, a concept known as incentive salience.
Incentive salience.
Right. The drug hijacked the reward circuitry to such a degree that the brain views cannabis not as a recreational choice, but as a critical survival need, on par with food or water.
It tags the drug with an overwhelming wanting signal.
It essentially installs a flashing neon survival sign in the brain, pointing directly at cannabis.
Wow. But in week 2, as the prefrontal cortex regains functional connectivity with the subcortical regions like the striatum and the amygdala, your executive control over that craving improves.
The PFC starts dampening that exaggerated incentive salience. The neon sign begins to dim.
But I want to push back on this slightly, or at least add a critical caveat. Because while the physical cravings decrease, week 2 is notoriously cited in addiction literature as a high relapse window.
It is.
People often report intense depression and anxiety peaking around day 10. If the cravings are less physical and the PFC is coming back online, why is the vulnerability to relapse still so dangerously high?
That is a vital nuance. The vulnerability remains high precisely because the withdrawal symptoms transition from the somatic to the affective.
Meaning from physical pain to emotional distress.
Exactly. The sweating and headaches are largely gone, but the baseline anxiety, the lingering hypodopaminergic anhedonia, and the depressive symptoms often peak. Because the individual is in a state of emotional distress, environmental cues become highly potent, dangerous triggers.
Let's trace the neural pathway of an environmental cue. You're feeling physically fine, but you have a highly stressful conflict at work. Or you simply walk into your living room and sit down on the exact spot on the couch where you historically consumed cannabis every evening at 6 p.m. What happens in the brain in that millisecond?
Your hippocampus, the memory center, recognizes the spatial context of the couch, or the emotional context of the work stress.
Right.
It immediately communicates with the amygdala, recalling the cannabis was the primary, highly effective tool previously used to soothe that specific environment or emotion.
The amygdala projects this signal rapidly to the striatum.
Which generates a sudden, intense psychological urge to use the drug.
It's a deeply ingrained mental habit loop, a conditioned reflex. The brain says, stress detected, execute soothing protocol.
Precisely. However, the key difference in week 2, compared to the acute phase, is the duration and the manageability of that cue-induced craving. Because your prefrontal cortex is recovering its top-down connectivity, you now have the biological braking power to intervene.
You can actually witness the thought forming.
Yes.
You can recognize the urge as a conditioned habit loop triggered by sitting on the couch, rather than a physiological mandate. You have the executive bandwidth to let the thought pass without translating it into motor action.
You are transitioning from being a passenger driven by deep physiological deficits to being the driver, relying on a recovering executive network to navigate the psychological triggers.
It is a profound shift from physical dependency to psychological behavioral management.
It really is.
The interconnectivity of all these mechanisms is staggering. The CB1 receptors upregulating across the cortex, the striatal dopamine synthesis normalizing to restore motivation, the REM sleep backlog processing emotional debt.
The prefrontal cortex shaking off its hypoglutamatergic suppression to regain executive control.
And the cravings shifting from physical demands to manageable cognitive loops. It paints an incredibly vivid, dynamic picture of what is happening inside the skull during days 7 to 14. But to truly appreciate the scale of this biological construction project, we need to zoom out. Let's look at the broader timeline. How does this critical week 2 window connect to the macroscopic picture of recovery?
Context is essential to understanding the full trajectory. If we map out the timeline starting from day 0, the moment of cessation, the first phase is the 0 to 3 day window.
The acute survival phase.
The opponent process theory dominates. The brain is profoundly shocked by the sudden lack of exogenous cannabinoids. This is characterized by the strongest somatic cravings, the highest irritability, debilitating insomnia, and severe gastrointestinal distress. The system is crashing.
Then we enter our primary focus period, the 7 to 14-day window.
Here, the acute physical crisis has largely passed, and the intense structural rebuilding begins. The CB1 receptors are actively migrating back to the cell surfaces.
Sleep architecture is improving in duration, though dominated by the intense REM rebound.
The profound anhedonia slowly begins to lift as natural motivation tentatively returns via stabilized dopamine synthesis.
And the deep cognitive fog begins to clear as the PFC regains connectivity.
Exactly.
And what happens as the individual pushes past this critical window? What does the biology look like at 3 to 4 weeks and beyond?
By the 3 to 4 week mark, major neurochemical balance has returned for the vast majority of individuals. The density of CB1 receptors across the cortical regions is almost entirely normalized to pre-exposure baselines.
The dopamine signaling and synthesis capacity are stable, meaning natural rewards are fully effective again.
The REM sleep debt is largely paid off and sleep architecture stabilizes.
Consequently, the higher-order cognitive functions, sustained attention, complex working memory, and optimal decision-making capabilities fully normalize.
It's a comprehensive reset.
One question that frequently comes up when looking at this timeline is the sheer length of it. If you look at the acute withdrawal timelines for many other substances, even highly physically addictive ones, the acute somatic phase is often over in a matter of days. And the brain seems to initiate recovery much faster. Why is the neurobiological timeline for cannabis cessation so uniquely protracted? Why does it take a full 4 weeks for the receptors to fully trust the new baseline?
The answer lies entirely in the specific pharmacokinetics of THC. THC and its primary metabolites are highly lipophilic.
Meaning they are exceptionally fat-soluble, they do not dissolve well in water.
Exactly. Unlike a substance like alcohol, which is water-soluble and cleared from the bloodstream and the central nervous system very rapidly.
Right.
When an individual consumes cannabis chronically, the THC isn't just acting on the brain, it is being actively absorbed and stored in the body's adipose tissue, the fat cells.
When the individual ceases consumption, those fat cells act as a massive reservoir.
They do. They slowly, continuously release the stored THC and its metabolites back into the bloodstream over time.
It leaches out in a slow, tapering trickle.
Precisely. This lipophilic storage and slow release mechanism is why THC metabolites can be detected in urinalysis for weeks, or in heavy chronic users, sometimes well over a month after the last instance of consumption.
And crucially, this explains why the neurobiological timeline is so protracted. The brain isn't dealing with a clean, sudden break.
No. Even though the individual has stopped consuming, their brain is still receiving a microscopic tapering trickle of the exogenous cannabinoids released from their own fat stores.
So the brain takes a full 3 to 4 weeks to hit those major restorative milestones because it takes that long for the systemic reservoir to truly drain.
Only then is the central nervous system completely clear, allowing the receptor networks to fully stabilize at their natural endogenous baseline without external interference.
It is an absolute masterclass in biological engineering. The way the system adapts, defends itself, and eventually rebuilds is awe-inspiring. Let's synthesize everything we've uncovered today. By dissecting this massive stack of clinical literature, imaging studies, and behavioral data, what we have fundamentally mapped out is the incredible resilience of the human central nervous system.
The capacity for neuroplasticity and self-correction is truly profound.
Within a mere 14 days, just 2 weeks of abstinence, we witness a biological construction project of staggering proportions. Receptors that were literally dismantled and pulled inside the cell bodies to hide from the constant overstimulation of THC are reassembled and pushed back to the neural surface.
The volume knobs of the cortex are reinstalled.
The chemical dopamine factories that were shut down find their natural rhythm again, allowing the reward system to finally remember how to find joy in the simple, everyday moments of life.
The massive sleep debt is aggressively audited and paid down through intense REM rebounds.
And perhaps most importantly, the executive center, the prefrontal cortex, wakes up from its hypoglutamatergic slumber, rewires its connections to the deeper brain, and takes back the steering wheel from the reactive, impulse-driven basal ganglia.
And understanding the exact mechanics of this 13-day timeline raises a final, very important consideration for us. When we, as a culture, discuss quitting a substance or breaking a deeply ingrained behavioral loop, society overwhelmingly frames it as a test of moral character. We reduce the entirety of addiction and recovery to a simple binary matter of willpower.
Right. The cultural narrative is always, just make a better choice, or just push through it. If you relapse, you just didn't want it badly enough.
But the clinical imaging, the high-resolution PET scans, the structural MRIs, the measurable neurotransmitter deficits, they tell a completely different, highly objective story.
They really do.
The scientific sources show us definitively that the first 2 weeks of quitting cannabis are not a moral struggle. They are a literal, mechanical rebuilding of physical brain infrastructure. You are regrowing receptor proteins, stabilizing complex neurotransmitter synthesis lines, and physically rewiring large-scale neural networks. It is a biological construction zone inside the skull.
So here's a provocative thought to leave you with. How might our approach to personal habits, to addiction, and to recovery fundamentally change if we fully embrace this science? What if we completely stopped judging people or judging ourselves for a perceived lack of willpower, and instead provided the patience, the accurate medical understanding, and the empathy required to support the complex biological construction project actually happening under the hood? What if we viewed the struggle of week 2 not as a test of moral strength, but as a period of necessary, physically demanding physiological healing?
Shifting that paradigm from moral failing to biological recovery would fundamentally alter the landscape of clinical treatment and personal self-compassion.
It absolutely would change everything. Well, that brings us to the end of our deep dive into the 7 to 14-day neurological timeline of quitting cannabis. We want to warmly thank you, the listener, for spending this time with us and exploring the incredible intricacies of the human brain.
It has been a true pleasure to unpack this complex science with you.
Keep asking the hard questions, keep looking under the hood of how things really work, and stay relentlessly curious. We will see you on the next deep dive.
When THC enters your brain, what actually happens inside your neurons? Is it relaxation, or is your brain slowly changing its reward system?
That is, well, that's the exact question we're tackling today.
Right. Welcome to another deep dive. Today, we are going on a journey, and the destination is actually your own mind. We're going to shrink down to a microscopic level to explore your neural architecture.
Which is, no small task.
Not at all. Our mission for this deep dive is to decode the exact molecular sequence of events that occurs the moment cannabis is consumed. Whether you're a regular consumer, someone who partakes occasionally, or just someone who's insanely curious about neuroscience, we're going to bypass the cultural noise entirely.
Because there's a lot of cultural noise out there.
So much of it.
Yeah.
But we're going to look strictly at the biological reality to understand how this plant interacts with your hardware.
And to do that properly, we've assembled a really massive, highly detailed stack of sources for today's deep dive. We are pulling from clinical research papers, comprehensive neurobiological reviews, positron emission tomography or PE imaging studies, and leading addiction medicine journals.
Yeah, those PE scans they're wild to read about.
They are. These sources meticulously detail the pharmacokinetics of cannabinoids, how they map onto human brain regions, and the exact timelines of the brain's recovery and adaptation.
I was actually looking at those PET scan studies in our source stack before we started. It's incredible that we can inject a safe radioactive tracer into a living human being, put them in a scanner, and literally watch their brain receptors light up on a screen.
Right? It takes all the guesswork out of it.
Yeah, exactly. We aren't just relying on people filling out surveys about how they feel. We're actually watching the molecular machinery of the brain adapt in real time.
That's the beauty of modern neuroimaging. I mean, for decades, the study of cannabis in the brain was heavily reliant on subjective reporting or observational behavioral studies. But the literature we're looking at today goes right down to the synaptic cleft.
It maps where the molecules go.
Where they go, how long they stay, and what structural changes they leave in their wake.
It's a mountain of data, but it tells a truly incredible story about how adaptable and dynamic your brain really is.
Yeah.
Okay, let's unpack this. Before we even talk about a plant, before we talk about smoke or vapor or edibles, we have to look at the baseline state of the human brain.
The baseline, yes.
We have to look at what your brain is doing right now, completely on its own, before any external substance is introduced. Because if I'm understanding the research correctly, the brain is not just sitting there waiting for cannabis to arrive.
Not at all. To understand how cannabis affects the brain, you first have to understand that your brain already has its own built-in cannabis-like network. It's called the Endocannabinoid System, or the ECS.
And it's old, right?
From an evolutionary perspective, it's ancient. It predates the cannabis sativa plant by about 575 million years in vertebrate evolution.
Let's pause there for a second because that number is staggering. 575 million years. That means long before humans existed, long before mammals existed, this biological network was already up and running.
It evolved in early vertebrates in the ocean. Sea squirts, early fish, amphibians, they all share this underlying biological architecture. Nature found a molecular communication system that worked incredibly well for regulating biological functions, and it kept it.
Long before humans were cultivating anything in the soil.
Exactly. This internal system was fundamental to the survival and functioning of complex nervous systems way before that.
So what exactly is this system doing in there? If we have our own internal cannabis system, what are the chemicals it's actually producing? I know we produce our own natural painkillers like endorphins, but what's the natural equivalent of cannabis?
The body produces two main endogenous cannabinoids. Endogenous simply meaning originating from within an organism. The first one scientists discovered is called anandamide.
Anandamide.
Yes. The name is actually derived from the Sanskrit word ananda, which translates to bliss or extreme happiness.
The bliss molecule. That's a pretty powerful name for a chemical produced by your own body.
It is, and it speaks to its profound effects on mood and well-being. But there's a second endogenous cannabinoid, which was discovered shortly after, and it's actually much more abundant in the brain. It's a molecule called 2-arachidonyl glycerol, but neuroscientists usually just abbreviate it to 2-AG.
Anandamide and 2-AG, the bliss molecule and its highly abundant partner, what's their day job? If they're floating around in your head right now, what are they actively doing for you as you sit there listening to this?
They're the master regulators of homeostasis in your body. Homeostasis is just the scientific term for biological balance. They regulate a massive spectrum of critical, everyday bodily functions. We're talking about your mood, your ability to form and forget memories, your stress responses, how you modulate pain, your sleep cycles, and your appetite.
That's a huge portfolio.
It really is. But what makes anandamide and 2-AG so entirely unique is how they function at the cellular level. They completely break the traditional rules of neuroscience.
Yeah, I noticed this when going through the neurobiological reviews, and honestly, it kind of broke my brain for a minute. Let's actually visualize this for the listener, because when we learn about the brain in high school biology, we learn a very specific linear model of how neurons talk to each other.
The classical understanding of neurotransmission is a one-way street. Imagine a microscopic gap between two nerve cells. The sending cell, the presynaptic neuron, stores its chemical messengers in tiny little microscopic bubbles called vesicles.
Like a warehouse full of packed boxes ready to ship.
Exactly. And those boxes are full of things we've all heard of, like dopamine for reward, or serotonin for mood, or glutamate to excite the next cell. When an electrical signal comes down the nerve fiber, it triggers those vesicles to fuse with the cell wall and dump their chemical contents across the gap, which is called the synapse. Those chemicals float across the gap and land on the receiving cell, the postsynaptic neuron, clicking into receptors like keys into locks.
The message is received, and the signal continues forward.
It's always forward communication, sending cell to receiving cell.
Okay, so that's the textbook model. A one-way street of packed boxes.
Yeah.
How do our natural endocannabinoids, anandamide and 2-AG, break that rule?
First, endocannabinoids do not sit in vesicles waiting to be fired. There's no warehouse of packed boxes. They are created on demand from the lipid membranes, the fatty outer layer of the brain cell itself, only at the exact millisecond they are needed.
So the cell literally synthesizes these bliss molecules out of its own fatty outer wall in real time. It just manifests them.
It does, and then it does something even more bizarre. It sends them completely backwards.
Oh.
Yes. If the receiving neuron is getting overstimulated, let's say it's getting blasted with way too much excitatory glutamate and it's starting to become chemically overwhelmed, it synthesizes 2-AG or anandamide on demand and pushes it backward across the synapse.
It swims upstream against the current.
Exactly. It travels retrograde, it goes back to the sending neuron, and it binds to specific receptors on that sending cell to tell it to quiet down. The primary receptors they bind to in the brain are called CB1 receptors.
Are there other types?
There are also CB2 receptors, but those are mostly found out in the immune system and peripheral tissues. For our cognitive deep dive today, the CB1 receptor is the main character.
I love thinking about it like a biological circuit breaker. Or let's use a simpler analogy. Imagine a volume knob on a stereo system. If the neural signal gets way too loud, chaotic, and distorted, the receiving cell sends this backward messenger to reach out and literally turn the volume knob down.
That's a great way to look at it.
It tells the sending cell, hey, we got the message, you're being too loud, you can stop shouting now. It keeps the whole internal network balanced, so nothing burns out or short circuits.
That is a perfect way to visualize it. And what's so compelling is that this subtle, backwards signaling volume control is responsible for things we used to attribute to entirely different mechanisms.
Right. I was reading the section in our sources about the runner's high, and this blew my mind. For decades, we were told that the feeling of euphoria, that zen-like state and pain relief you get after running 5 miles, was all due to endorphins.
That was the prevailing theory for a very long time. Endorphins are the body's natural opioids. But recent research has shown that endorphins are actually quite large molecules. They have a very hard time crossing the blood-brain barrier.
So while your muscles might be swimming in endorphins after a run, your brain isn't necessarily getting them.
Right, so if it isn't endorphins giving you that mental high, what is it? It's the endocannabinoid system. Anandamide specifically.
The bliss molecule.
Exactly. When you put your body through the prolonged physical stress of running, your body starts synthesizing anandamide on demand. Because it is a highly fat-soluble lipid, it easily crosses the blood-brain barrier. It floods into your central nervous system, binding to those CB1 receptors, turning down the volume knob on pain signals, reducing your physiological stress response, and producing that profound, floating sense of well-being.
So that elusive runner's high is quite literally a natural internal cannabis high. It's your body manufacturing its own bliss to keep you moving forward. Your brain already produces its own cannabis-like chemicals to keep the system balanced.
What's fascinating here is how perfectly the chemical structure of the cannabis sativa plant mimics this ancient, delicate biological system. Nature essentially evolved a plant that produces a skeleton key for your brain's internal volume knobs.
Let's bring that skeleton key into the picture. We've established this beautiful, delicately balanced backward swimming system of anandamide and 2-AG. But then, a person decides to consume cannabis. Whether they're smoking it, vaping it, or eating an edible, the primary psychoactive molecule entering their bloodstream and eventually crossing the blood-brain barrier is delta-9 tetrahydrocannabinol, THC.
The main active ingredient.
Right. What happens the second THC meets this delicate endocannabinoid system?
To the brain, the molecular structure of THC looks incredibly similar to anandamide and 2-AG. It's a plant-derived lipid, a phytocannabinoid. It has a 21-carbon terpenophenolic skeleton that happens to be the exact perfect geometric shape to slide directly into those CB1 receptors we just talked about.
It's biological mimicry.
Precisely. It's a molecular imposter. And remember, CB1 receptors are not just sprinkled randomly throughout your head. They are incredibly dense in highly specific, highly evolved areas of the human brain.
Location is everything in neuroscience. Where exactly are these CB1 locks concentrated? If THC is the key, what specific doors is it unlocking?
They're heavily localized in three main areas. First, the cerebral cortex, which is the wrinkled outer layer of your brain that governs higher cognitive function, sensory perception, and complex thought.
Okay, the thinking center.
Second, they're extremely dense in the hippocampus, a seahorse-shaped structure deep in the brain that is the absolute epicenter for memory formation and spatial navigation. And third, they are packed into the basal ganglia and the cerebellum, which are the regions that control motor coordination, movement, and habit formation.
So when you introduce THC, you are sending this molecular imposter straight into the exact neural command centers that handle deep philosophical thinking, making memories, and physical coordination.
Straight into the core systems.
But you mentioned earlier that our natural endocannabinoids like 2-AG are released on demand in tiny, very specific locations just to quiet down one loud synapse. How does a hit of THC compare to that natural pinpoint release?
It's the difference between a targeted private text message and a massive blaring public address system. Natural endocannabinoids are released exactly where they are needed, in minuscule amounts, just to tweak the volume on one specific overactive neuron. And as soon as they do their job, the brain has a cleanup crew waiting.
A cleanup crew?
Yes, metabolic enzymes, specifically one called FAAH fatty acid amide hydrolase, which breaks down anandamide, and another called MAGL, which breaks down 2-AG. The moment the natural cannabinoid delivers its message to turn down the volume, these enzymes swoop in, chop the molecule up, and clear the synapse. The signal is incredibly short, highly precise, and tightly controlled by the brain's internal logic.
But THC doesn't play by those rules, does it? The brain's cleanup crew doesn't know what to do with it.
Not at all. THC floods the entire brain simultaneously. It binds indiscriminately to CB1 receptors everywhere at once in the cortex, the hippocampus, the basal ganglia, all at the same time. Furthermore, because it's a plant-based molecule and slightly different in its atomic makeup, it is not broken down quickly by those FAAH or ANGL enzymes.
So it just sits there, holding the volume knob down.
Exactly. And because THC is highly lipophilic, meaning it loves fat, it gets sequestered in the fatty cell membranes and tissues of the brain. It lingers. Instead of a signal that lasts for milliseconds, the THC signal lasts for hours, or even days and weeks in the fatty tissues of chronic users.
Let's expand on an analogy here, because this is crucial to understanding the subjective experience. THC is like a fake key that fits perfectly into the brain's cannabinoid locks. It opens doors that were never meant to be opened all at once.
I like that.
Imagine your home has this incredibly smart, carefully balanced internal thermostat. It has sensors in every single room. If the kitchen gets a little too hot because you're cooking, the system subtly drops the temperature just in the kitchen. If the bedroom gets a little chilly at night, it warms up just the bedroom. It's constantly making micro adjustments, so you never even notice it. You're just perfectly comfortable.
That is a very accurate representation of the natural endocannabinoid system maintaining homeostasis.
Right. But then an external force, THC, hacks into the mainframe. It completely overrides the smart system. It cranks the heat up to maximum in the living room, shuts the air conditioning completely off in the kitchen, turns on all the humidifiers, and completely bypasses the safety sensors that are trying to shut it down. The system is hijacked.
Hijacked is the right word.
That profound system-wide override where multiple brain regions are simultaneously having their volume knobs forcibly turned down against their will is the biological reality of what you experience subjectively as feeling high.
And nowhere is that override more impactful and potentially more concerning in the long term than in the brain's reward loop. If we want to understand motivation, desire, and why cannabis can become a deeply ingrained habit for some people, we have to look very closely at the mesolimbic dopamine system.
Whenever people talk about rewards, motivation, or addiction, dopamine is usually the first word that gets thrown around. But based on these papers, it seems like we often misunderstand how dopamine actually works. It's not just a pleasure chemical that makes you feel happy, is it? Let's actually map this out for the listener. Where's this reward loop, and what does it actually do?
You're absolutely right that dopamine is misunderstood. It's less about pleasure and much more about anticipation and motivation. The mesolimbic pathway is basically a neural superhighway. Let's visualize it. Imagine the very center, the most primal, ancient part of your brain, deep near the brainstem, that is the midbrain. Deep in there is a tiny cluster of cells called the ventral tegmental area, or VPA. Think of the VTA as the engine room of your desires.
Okay, I have the engine room in the deep midbrain.
Now, imagine a superhighway leading from that engine room straight up to the front of your brain, right behind your forehead, to a cluster of neurons called the nucleus accumbens. This circuit is your brain's evolutionary motivation center. It's designed to make you feel driven to do things that promote survival. Eating a calorie-rich meal, socializing with your tribe, reproducing. When you do these things, the VTA sends dopamine down the highway to the nucleus accumbens.
When the dopamine hits the nucleus accumbens, it acts like a highlighter pen.
Exactly. It assigns what neuroscientists call incentive salience to the stimulus. It highlights the behavior in neon lights and tells your brain, this is extremely important for our survival. Pay attention to how you did this and do it again.
But the brain doesn't just let dopamine flow freely all the time. If it did, we'd be manic, constantly chasing every single impulse. There has to be a brake pedal on this engine.
And there is. Left to its own devices, the dopamine neurons in the VTA would fire constantly and chaotically. So the brain keeps them tightly in check using a network of inhibitory interneurons that release a chemical called GABA. GABA is the brain's primary calming neurotransmitter. These GABA neurons act like a biological parking brake clamped onto the dopamine engine.
So the GABA is constantly pulling the reins back, saying, whoa, slow down, save the dopamine for something that actually matters.
Precisely. They ensure that your dopamine neurons only maintain a low, steady hum. We call this tonic firing. It runs at about 1 to 5 hertz, meaning the neurons are firing 1 to 5 times per second. It's just enough baseline dopamine to keep you out of bed, engaged with life, and mildly curious, but not overwhelmed.
Okay, so we have the VTA engine room, the dopamine highway leading to the nucleus accumbens, and the GABA brake pedal, keeping everything at a nice, steady 1 to 5 hertz hum. Enter THC. How does this molecular imposter hack the reward system?
It comes down to those CB1 receptors again. The GABA interneurons, the brakes, are absolutely covered in CB1 receptors. When THC floods the brain, it binds to these receptors on the brake pedal. And remember what endocannabinoids do? They are retrograde messengers that turn down the volume. They tell the cell to stop releasing its neurotransmitter. So when THC binds to the GABA cell, it forces it to stop releasing GABA.
Wait, so THC stops the release of the calming chemical. It inhibits the inhibitor.
Yes. The technical term for this in neuroscience is disinhibition. By shutting off the GABA interneurons, THC essentially takes a pair of wire cutters and cuts the brake lines on your dopamine system. Without GABA holding them back, the dopamine neurons in the VTA suddenly slip the leash. They switch from that quiet, steady 1 to 5 hertz tonic hum to rapid, explosive bursts. This is called phasic firing, and the neurons start firing at 20 hertz or more.
So they go from a slow heartbeat to a full-on sprint.
Exactly, and this causes a massive unnatural surge of dopamine to dump into the nucleus accumbens all at once.
And your brain, doing exactly what it evolved to do over millions of years, registers that massive dopamine surge as a monumental reward. It feels like you just accomplished something incredibly important for your survival, like you just secured food for the winter or won a massive victory, even if you were literally just sitting on the couch staring at a wall. The brain learns this chemical shortcut gives pleasure faster than normal life.
Which feels great in the moment.
Right. But this is where the dark side of homeostasis comes in. If we connect this to the bigger picture, the brain does not like being tricked. It notices when the engine is being redlined, doesn't it?
It notices immediately. The brain's ultimate goal is always to protect its balance. It hates being pushed out of its normal operating range. If you expose the brain to chronic, heavy doses of THC day after day, week after week, it realizes that the dopamine system is constantly being artificially stimulated. So to protect itself from this toxic overstimulation, it begins to adapt.
How does it adapt? Does it build stronger brakes?
No, it goes after the dopamine itself. It starts down-regulating its own natural dopamine system. It signals the VTA to produce less baseline dopamine, and it reduces the physical availability of dopamine receptors in the nucleus accumbens. It essentially packs away the receptors so they can't be overstimulated anymore.
It creates a hypodopaminergic state. Hypo, meaning low. So your baseline drops below normal. Let's talk about what that actually means for the listener in their daily life. If your baseline dopamine system is suppressed because the brain is trying to defend itself against the THC floods, how does that translate to your behavior, your mood, and your personality?
This is where we see the clinical manifestation of what has historically been called amotivational syndrome. It presents as profound apathy. More clinically, it leads to a state called anhedonia. Anhedonia is the inability to feel pleasure from normal, non-drug rewards.
Because the dopamine system has been dulled, everyday joys simply don't register on the scale anymore.
Exactly. Think about the normal things that give a healthy brain a little bump of dopamine. A great, engaging conversation with a close friend. eating a delicious meal at your favorite restaurant, watching a beautiful sunset, or the deep satisfaction of achieving a hard-fought goal at work or school. In a hypodopaminergic state, those gentle, natural stimuli fail to produce enough signal to stimulate that blunted reward pathway. You experience them, but you don't feel the joy of them. The world becomes a bit gray.
The bar for feeling good has been artificially raised so high by the THC that normal life can't clear it.
Precisely. The only thing that provides a sufficient dopamine spike to feel normal or to feel motivated enough to do the laundry or run errands is introducing more THC to cut the brakes again. The motivation to engage with life is outsourced to a plant.
That's a chilling realization. The brain reorganizes its entire priority list around that chemical shortcut. But dopamine and motivation are just one piece of the puzzle. Let's shift away from the reward center and talk about the areas of the brain that govern how we actually think, decide, and remember. Let's talk about the prefrontal cortex and the hippocampus, because the source material goes into incredible detail here about executive function.
The prefrontal cortex, or PFC, is arguably the most highly evolved part of the human brain. It's a large region sitting right behind your forehead. This is your command center for executive function. It is responsible for sustained focus, complex decision-making, impulse control, and long-term planning. It's the part of your brain that allows you to weigh the future consequences of your present actions.
I always think of the prefrontal cortex as the adult in the room. It's the angel on your shoulder telling you to study for the test tomorrow instead of watching another 3 hours of television. It's the filter that stops you from saying something inappropriate to your boss. How does THC interact with this command center?
In the prefrontal cortex, the primary neurotransmitter involved is not GABA or dopamine, but glutamate. Whereas GABA is the brain's main calming chemical, glutamate is the brain's main excitatory neurotransmitter. It's the gas pedal for cognitive processing. It makes neurons fire and pass complex information along.
Okay, so glutamate is the spark of thought.
Yes, and just like we saw with GABA, the prefrontal cortex has CB1 receptors. When THC binds to these receptors in the PFC, it heavily suppresses the release of glutamate. By suppressing glutamate, THC plunges the prefrontal cortex into a hypoglutamatergic state.
Low glutamate, so the spark is dimmed.
Exactly. The complex neural networks that support executive control are literally deprived of the excitatory signals they need to function optimally.
So your ability to step back, reflect, and regulate your own behavior is severely impaired. The adult in the room is gagged, the top-down control weakens, and your behavior shifts dramatically toward impulsivity, risk-taking, and short-term gratification. The very part of the brain that controls discipline becomes less active. And our sources point out that this is particularly dangerous for adolescents, right? It isn't just a temporary dimming, it can actually change the physical structure of the brain.
Crucially so. The prefrontal cortex is one of the last areas of the brain to fully mature. It undergoes massive pruning and reorganization during the teenage years, continuing to develop well into a person's mid-20s. Exposing this actively developing circuitry to chronic THC has lasting structural consequences. Structural MRI studies highlight a specific subregion called the medial orbital prefrontal cortex, or MOPFC.
The medial orbital prefrontal cortex, what does that specific patch of brain do?
It's heavily involved in reward valuation and integrating emotion into decision-making. In adolescents who abuse cannabis heavily, the MRI imaging shows a significant, measurable decrease in the physical volume of the MOPFC.
A literal physical reduction in brain volume in the decision-making center. It shrinks.
Yes, the gray matter volume is reduced. And this is not just an anatomical quirk, it has real-world consequences. The literature details behavioral tests, like the Iowa gambling task. This is a psychological assessment used to simulate real-life decision-making. Participants are given 4 decks of cards and have to figure out which decks yield long-term profits and which lead to steep losses.
So it tests their ability to spot a bad deal and adjust their strategy.
Exactly. And the data directly correlates the reduced volume in the MOPFC with poor performance on this task. Cannabis-abusing teens show poor judgment, diminished choice optimization, and a marked inability to anticipate future outcomes. They keep choosing the risky, high-loss decks because their brains are structurally less capable of integrating the long-term consequences of their choices. The physical hardware required to foresee the future is altered.
That is incredibly sobering. It's one thing to feel lazy for a few hours. It's another to physically alter the architecture of your decision-making processes. But what about memory? We all know the cultural trope of the forgetful cannabis user, the person who walks into a room and completely forgets why they're there or loses their train of thought in the middle of a sentence. What is the actual biology behind that memory gap? Is it the same glutamate suppression?
It is related, but to understand memory, we have to look at the hippocampus. As we mentioned, this is the seahorse-shaped structure deep in the temporal lobe. The hippocampus is absolutely packed with CB1 receptors. To understand how THC disrupts it, we first need to know how a memory is actually made. When we learn something new, a name, a phone number, a route to a new restaurant, neurons in the hippocampus form stronger physical and chemical connections with each other.
They build a bridge.
Exactly. This cellular mechanism of building the bridge is called long-term potentiation, or LTP. It's the physical process of forging a memory trace. It requires a robust release of glutamate to activate specific receptors that tell the cell, grow stronger connections here.
I was looking at the electrophysiology studies in our source stack, and if I'm understanding the research correctly, it's not that the memories are being erased like a deleted hard drive. It's that the record button is literally jammed by the THC. Can we break down how that block actually happens physically?
You have it exactly right. THC directly blocks long-term potentiation. Because THC binds to the CD1 receptors and suppresses neurotransmitter release, it prevents the receiving neuron from ever reaching the electrical activation threshold necessary to forge that new memory bridge. The information comes in through your senses, but the biological concrete never sets. The memory trace simply vanishes.
So you experience an event, but your brain cannot physically write it down to the hard drive. But it isn't just about long-term memory, like remembering what you did yesterday. It's also about keeping a straight thought right in the moment, the working memory.
Right, and that relates to brainwaves. The hippocampus doesn't just pass chemicals back and forth. It relies on specific rhythmic pulses of electrical activity to process information. The most important of these for memory and attention are called gamma oscillations. These are fast brain waves that operate between 30 and 90 hertz.
30 to 90 pulses per second.
Yes. Think of gamma oscillations as the conductor of a massive symphony orchestra. You have millions of neurons, the musicians, and the gamma wave is the conductor keeping them all firing in perfect rhythmic synchrony. This synchrony is what binds different pieces of sensory information together so your thoughts remain organized and coherent.
And when THC floods the hippocampus, it effectively fires the conductor.
It creates chaos in the orchestra. THC disrupts the delicate balance of excitation and inhibition that the brain needs to maintain those rhythms. The gamma oscillations become fragmented and disorganized. The violins are playing at a different tempo than the cellos. Behaviorally, you experience this fragmented brainwave activity as a fragmented stream of thought.
You walk into a room and forget why. You're telling a story and suddenly stop looking at your friend saying, wait, what was I just talking about? The temporal framework for organizing information in your mind is temporarily shattered because the electrical rhythm has collapsed.
It is a profound disruption of the self in time and space.
And we have to address the elephant in the room regarding all of these cognitive impacts because it makes everything we just talked about significantly more urgent. Modern potency. We're not talking about the cannabis of the 1960s or 1970s. Historically, the cannabis plant yielded THC concentrations of maybe 2 to 4%. It was a relatively weak interaction with the endocannabinoid system.
That is correct. The historical data shows a very mild pharmacological profile compared to today.
But today, with advanced agricultural genetics, indoor hydroponics, and complex chemical extraction methods, modern vaping products, wax dabs, and concentrated edibles can reach THC concentrations of upwards of 80 to 90%. We are flooding these ancient, delicate endocannabinoid receptors with a chemical payload that is magnitudes higher than anything the human brain has ever encountered in its 575 million years of evolutionary history.
The pharmacological load is unprecedented, and because THC is dose-dependent, these ultra-high concentrations dramatically amplify every single cognitive, structural, and dopaminergic impact we have discussed. The suppression of glutamate is deeper. The fragmentation of gamma waves is more severe. The downregulation of dopamine receptors happens faster and more profoundly.
It paints a stark, somewhat intimidating picture of the neurobiological toll of chronic use. We have essentially mapped out a system under siege. But there's a crucial transition we need to make here because this deep dive is not a tragedy. Our sources do not just document the disruption caused by THC, they meticulously detail the recovery. The brain is incredibly resilient. It is highly plastic. It actively works to rebalance and heal itself the moment THC is removed from the system.
This is perhaps the most important data in the entire stack of sources. Neuroplasticity, the brain's ability to reorganize itself by forming new neural connections, is a powerful force.
Let's talk about the timeline of that neuroplasticity and recovery. When someone who has been a heavy, chronic user decides to quit, What actually happens in those first few days? Because anyone who has tried to quit knows it is rarely easy. There's a physiological hurdle to get over first.
The initial hurdle is clinically defined as cannabis withdrawal syndrome, or CWS. For a very long time in popular culture, there was a misconception that cannabis was just a plant and didn't have physical withdrawal symptoms like alcohol or opiates. But biologically, withdrawal is simply the brain's opponent processes left unopposed.
Opponent processes. Let's dig into that term. What does that mean in the context of the brain?
Imagine a massive game of tug-of-war. On one side, you have THC pulling hard on the rope. It is suppressing glutamate, suppressing GABA, turning down the volume knobs everywhere, and artificially sedating the nervous system. The brain, whose only goal is homeostasis, wants balance. So it recruits its own team and pulls back hard in the opposite direction. It starts pushing to produce more excitatory neurotransmitters to overcome the heavy suppression of the THC. It's pulling back with all its might to keep you awake and functioning.
So the brain is fighting the drug. But what happens when you suddenly stop using THC? You drop your end of the rope.
Exactly. When you quit cold turkey, THC drops the rope. But the brain doesn't know the game is over instantly. It's still pulling backward with all its might.
So the brain's countermeasures are suddenly running totally unopposed.
Yes. Without the heavy blanket of THC suppressing the system, there is a sudden, massive rebound spike in glutamate and other excitatory chemicals. The volume knobs get cranked all the way up because the negative pressure is gone. This neurochemical volatility peaks in the first week of abstinence.
So what does that feel like subjectively? If your brain is suddenly swamped with excitatory chemicals running unopposed, how do you feel?
Behaviorally, you experience this as severe insomnia, intense irritability, generalized anxiety, and physical restlessness. You might sweat more or lose your appetite entirely because the endocannabinoid system that regulates hunger is completely dysregulated.
And the dreams. I was reading the sleep architecture studies, and this is fascinating. People report having the most intense, vivid, sometimes terrifying dreams when they quit. Why does that happen?
That is REM rebound. THC is known to suppress REM sleep, rapid eye movement sleep, which is the phase of sleep where we dream most vividly and process emotional memories. When you use THC heavily, you deprive yourself of normal REM cycles. When you quit, the brain tries to make up for lost time. The REM sleep rebounds aggressively, leading to a flood of vivid, intense dreaming as the brain attempts to process a backlog of emotional data.
It sounds incredibly uncomfortable, but framing it as an opponent process is actually really encouraging. It means the discomfort is literally the sound of the engine restarting. It's the biological proof that the brain is actively recalibrating and fighting its way back to homeostasis.
It is the pain of healing.
And what about those CB1 receptors? We talked earlier about how the brain downregulates them, essentially packing the locks away and hiding them from the massive floods of THC to protect the dopamine system. If you quit, do those receptors ever come back to the surface?
They do, and the timeline is one of the most amazing findings in modern neuroimaging. Using those PET scans you mentioned at the beginning, which allow researchers to inject radio tracers that bind to CB1 receptors and literally visualize receptor density of living human brains, studies have meticulously tracked this recovery. Within just 48 hours, 2 days of abstinence, the downregulated CB1 receptors begin to return to the surface of the neurons. They start upregulating.
2 days. That's incredibly fast for a biological system that has been suppressed for months or years to start rebuilding itself.
It is remarkably fast, and it continues steadily. The PET scan data from our sources shows that after 4 weeks of monitored abstinence, the CB1 receptor density in heavy chronic cannabis users becomes practically indistinguishable from that of healthy control subjects who have never used cannabis in their entire lives. The receptors rebuild, the locks return to the doors, the volume knobs are reinstalled.
That is phenomenal. It's a full structural reset of the receptor network in a month. But does absolutely everything recover in 4 weeks? What about the dopamine system and that lingering brain fog? Because anecdotally, people say it takes much longer to feel totally normal and motivated again.
They are right. While the CB1 receptors bounce back quickly, the downstream systems take longer to heal. Dopamine homeostasis takes a bit more time. The reward system is deeply entrenched, and restoring that baseline tonic firing, reversing the hypodopaminergic state, usually takes roughly 2 to 3 months of sustained abstinence.
Months 2 and 3. That seems to be the critical window.
It is. This is the period where the profound brain fog truly lifts. During these months, clinical assessments show that verbal memory significantly improves, attention span lengthens, and executive function in the prefrontal cortex stabilizes. The baseline dopamine levels rise back to their healthy 1 to 5 hertz hum.
And as that baseline rises, the mesolimbic pathway starts responding to natural joys again. That heavy anhedonia fades away. You start feeling genuine pleasure from a good meal, from exercise, from hanging out with your friends, or from achieving a goal. The color comes back into the world. Your motivation returns because the physical, biological hardware driving it has been repaired and polished. The brain is not broken, it's adaptive. When THC leaves the system, the brain begins rebuilding.
If we synthesize this entire journey, it is a profound testament to the power of homeostasis. We started with an ancient, delicate, deeply complex natural endocannabinoid system, a network of anandamide and 2-AG signaling backwards across synapses to maintain perfect, quiet harmony in our mood, memory, and metabolism.
A system that's been humming along for 575 million years.
Yes. We saw how THC, acting as an overwhelming chemical mimic, a molecular imposter, forcefully hijacks that system. It floods the receptors, cuts the GABA brakes on the dopamine engine to create a massive artificial reward, and plunges our prefrontal executive control centers into a suppressed, impulsive hypoglutamatergic state.
We saw the orchestra conductor fired, scattering our gamma waves and fragmenting our memories.
We saw the brain adapt by shutting down its own receptors and blunting its own joy to survive the toxic flood. And finally, we witnessed the brain's remarkable, relentless capacity to heal, to upregulate its receptors, to clear the fog, and to find its delicate balance once more when given the chance.
It's an epic biological narrative happening right inside our skulls. But before we wrap up, I want to leave you with one final provocative thought, something to mull over on your own that builds on everything we've just discussed from a slightly different angle.
This raised an important question for me when reviewing the literature, particularly the botanical and evolutionary data. The cannabis sativa plant is a botanical anomaly. We have spent this entire deep dive talking about THC, the psychoactive driver, the molecular key that hacks the reward system. But THC is not the only cannabinoid the plant produces. It also produces cannabidiol, or CBD, and CBD's interaction with the brain is a complete biological paradox.
Right. CBD has become incredibly popular in the wellness space. You see it in everything from tinctures to lotions. But from a neurological standpoint, how does it actually work in this context alongside THC?
Unlike THC, CBD has a very low affinity for CB1 receptors. It does not fit the lock perfectly. It does not bind to them like a fake key, and therefore it does not cause any intoxicating high whatsoever. But more than that, neurobiological studies suggest that CBD acts as what is called a negative allosteric modulator of the CB1 receptor.
Negative allosteric modulator. Let's translate that. What does it actually do to the receptor?
Imagine the CB1 receptor as a keyhole. THC is the fake key trying to go straight in. CBD doesn't try to go into the keyhole. Instead, it binds to the side of the lock and subtly changes the shape of the entire mechanism. By changing the shape of the lock, it makes it much harder for THC to bind and exert its extreme effects.
It acts like a buffer.
It does. In clinical studies, CBD actually appears to protect the brain from THC-induced damage. It has been shown to mitigate the memory impairment in the hippocampus, reduce the anxiety that high-potency THC can trigger in the amygdala, and even protect against the psychotic-like symptoms and extreme dopamine dysregulation.
So let me get this straight. You have THC, which dysregulates the dopamine system, fragments memory, shrinks the prefrontal cortex over time, and hacks the reward loop. And right next to it, growing on the exact same plant, synthesizing in the exact same glandular trichomes, you have CBD, a compound that actively acts as a neuroprotector against those exact mechanisms.
Exactly. It leaves us to wonder about the evolutionary biology of the plant itself. Why did nature package a powerful neural hijacker and its own specific, perfectly tailored neuroprotector inside the exact same leaf? In historical cannabis strains, the ones growing wild for thousands of years, these two compounds were relatively balanced. The plant naturally provided its own antidote, its own buffer to protect the brain's homeostasis.
But in the modern, hyper-cultivated strains designed in laboratories solely for maximum psychoactive impact, that natural protective ratio has been completely obliterated. We've bred the CBD out of the plant to make room for 90% THC. We've stripped away the biological shield to deliver a sharper sword.
It's a fascinating paradox. The plant contains both the chaos and the cure.
But ultimately, how that plant affects your life, your motivation, your memory, and your future isn't up to the plant. It isn't up to the genetics of the leaf. It is a choice about neurobiology. It's a choice about how you want your dopamine system calibrated and who you want driving the car. So the real question is not what THC does to the brain, but how much control we want to give it. Imagine your brain not just as an organ or a biological computer, but as this massive, sprawling, bustling metropolis. And in this incredibly dense city, you have roughly 86 billion residents.
86 billion. It's a staggering number when you really sit with it.
Right. And these residents, they're your neurons. They are constantly talking to each other, coordinating literally every thought, every heartbeat, every fleeting memory, every movement. And they do this using a highly specific lightning-fast electrical language. It is a city of perfect order, governed by strict traffic lights and precise communication lines.
Very delicate balance.
Exactly. But what happens when a single chemical compound derived from a plant, specifically delta-9 tetrahydrocannabinol, or THC, from cannabis sativa, floods into this city? Suddenly the traffic lights change. The communication lines are completely rewired. And even your very perception of the flow of time itself begins to stretch and warp.
It truly is a profound biological takeover. And the reason it works so seamlessly is because this plant compound is essentially picking a highly sophisticated lock that already exists within the human brain. The lock was never designed for the plant. The plant just happened to evolve a molecular key that fits it perfectly.
I love that visual. A master key that just happened to grow on a plant. And that is exactly the mission of our deep dive today. We are going straight into the source material to explore the profound, intricate neurobiological journey of THC in the human brain.
Yeah, and we're not just going to talk about what happens during a high in some vague, abstract sense. We are going to really explore the microscopic architecture of neural communication.
We'll look at how cannabis literally hacks an ancient biological circuit breaker. We will examine the neuroadaptations that occur with chronic use, and I think most importantly, we're going to trace the incredible timeline of homeostatic recovery when the brain is left to heal and recalibrate.
It's a journey that takes us from the absolute baseline of how a single neuron fires all the way to the complex cognitive restoration of executive functions. And to do this, we'll be looking closely at some fascinating studies. We've got everything from high-resolution KT imaging of the brain's receptors to detailed behavioral assessments, like classical eye blink conditioning.
We have a ton of ground to cover. So whether you are intensely curious about neurobiology, perhaps you're studying addiction, or maybe you just want to know exactly why time seems to slow down to a crawl when someone consumes cannabis, this deep dive is going to give you the complete picture. We're taking you from the very first action potential to the final stages of cognitive restoration.
To truly appreciate the magnitude of the THC hack, we first have to establish what normal looks like. We have to look at the baseline brain and really understand the foundations of the action potential.
Right, because every single function of the brain relies on neurons communicating across a tiny gap called the synapse.
Exactly. But before a chemical message can even be sent across that gap, an electrical spark has to travel down the neuron itself.
I was actually amazed looking at the research on how neurons rest. I always pictured them just sort of sleeping or sitting dormant when not in use. But they aren't dormant at all, are they? The neuronal membrane is described as this highly regulated, incredibly selective barrier.
Yeah, the term resting state is almost a misnomer because it implies a lack of activity. In reality, inside the cell, there is a negative charge of exactly negative 70 millivolts compared to the outside.
Negative 70 millivolts.
Right, and this negative charge is not some biological accident. It is a state of intense, carefully maintained readiness.
It's like a drawn bowstring, just waiting for the archer to let go.
That is the perfect analogy. And keeping that bowstring drawn requires a massive amount of energy. That state of readiness is maintained by one of the absolute hardest working mechanisms in the body. It's called the sodium-potassium pump.
The housekeeper.
The housekeeper, yes. Or maybe the power grid manager of our cellular metropolis. It constantly uses ATP, which is the primary energy currency of the cell, to actively pump 3 sodium ions out of the neuron for every 2 potassium ions it brings in.
So it's constantly kicking out more positive charge than it's letting in, which is exactly how the inside stays negative at that negative 70 millivolts. But the amount of energy that takes, I read that something like 20 to 40% of the brain's entire energy budget goes just to this one pump, just to maintain the resting state.
It is staggering when you think about it. The brain consumes about 20% of the body's total energy, and a huge fraction of that is just spent keeping these billions of bowstrings pulled back, working against the natural flow of diffusion.
Okay, let's unpack this, because the moment of action is where it gets explosive. When this neuron receives excitatory signals from its neighbors, the voltage starts to rise, right? It creeps up from that negative 70 millivolts until it hits a very specific threshold.
Yes, usually around negative 55 millivolts. And the second it hits that specific threshold, it's like a dam breaking. Voltage-gated sodium channels along the membrane just fly open.
And all that sodium that the housekeeper pump has been painstakingly pushing out suddenly rushes back in.
Because it wants to balance out the charge. Both the electrical gradient and the concentration gradient are pulling it inside, and this is depolarization.
Depolarization.
Got it. Precisely. The rush of positive sodium ions spikes the internal voltage of the cell all the way up to positive 30 or even positive 40 millivolts in just a fraction of a millisecond. That is the actual spark. That's the action potential.
But it can't stay there.
No, the neuron cannot stay in that highly positive state. Almost immediately, those sodium channels slam shut and a different set of doors opens, the potassium channels. Potassium, which is heavily concentrated inside the cell, rushes out, carrying its positive charge away to restore the negative environment.
Repolarization.
Exactly. And it even overshoots for a moment, dropping below negative 70 in a phase called hyperpolarization before the housekeeper pump kicks back into high gear to sweep the sodium back out and reset the whole system.
And this entire cycle, this spark, is an all-or-none event, correct? Like flipping a light switch. There is no such thing as a half-fire.
That's right. It fires or it doesn't. And the reason this microscopic electrical storm matters is because this action potential travels down the length of the neuron's long cable-like axon until it reaches the terminal.
And that's where the chemistry takes over.
Yes. Once there, the electrical signal triggers the influx of calcium. Calcium acts as the final trigger, forcing these tiny bubbles, called vesicles, which are full of chemical neurotransmitters, to fuse with the membrane and spill their contents into the synapse, that gap between neurons.
So the electrical spark inside the city's wires turns into a chemical ferry crossing the river to the next building.
Beautifully put. This chemical message floats across the gap and binds to receptors on the next neuron, telling it to either fire its own action potential or stay quiet. This is the forward-moving, highly polarized language of the brain. It is literally how you think, how you move, and how you feel.
But the brain is incredibly dense. The city is packed. If neurons were just constantly firing these action potentials, shouting at each other with excitatory neurotransmitters like glutamate, wouldn't the whole system just overheat? I mean, it seems like it would be wildly overstimulated. There has to be a way to regulate this constant firing so the city doesn't literally short-circuit.
And that profound need for regulation brings us to the discovery of the Endocannabinoid System, or the ECS. When researchers first started piecing the system together, they realized they were looking at a master regulator of homeostasis. Its entire evolutionary purpose is to keep the physiological systems of the brain and body within a very narrow, safe, healthy operating range.
This is one of my absolute favorite concepts from the source material. Because normally, as we just established, the flow of traffic is strictly one way. Neuron A sends a message forward to neuron B. But the ECS operates entirely in reverse. It utilizes something called retrograde signaling.
What's fascinating here is that for the longest time, the scientific consensus, the absolute dogma in neuroscience, was that synaptic transmission was strictly a one-way street, forward only.
Right, nobody even considered backward signaling.
Exactly. But researchers in the early 1990s studying regions like the hippocampus and the cerebellum were completely stunned. They discovered that neuron B, the receiving neuron, could actually talk back to neuron A to shut it down.
It was like watching cars successfully drive backward on a one-way highway. So how exactly does neuron B do that? Let's say neuron B is getting absolutely hammered by excitatory signals. Neuron A is just shouting glutamate at it nonstop. To protect itself from overheating, neuron B creates endocannabinoids directly on demand, right from the fat, the lipids in its own cell membrane.
Yes, and that on-demand aspect is crucial. They aren't stored in little vesicles waiting to be released like normal neurotransmitters. Because they are lipids, fat-soluble, they would just slip right through the vesicle walls anyway. They are synthesized exactly when and where they are needed.
And what are these molecules called?
The two main endocannabinoids your brain produces are 2-arachidonylglycerol, which we usually just call 2-AG, and anandamide.
I love the history behind that naming. Anandamide gets its name from the Sanskrit word ananda, which translates to bliss or divine joy. It's the bliss molecule.
It is a very fitting name for a molecule that induces relaxation and balance. Once neuron B synthesizes these endocannabinoids, because they are fat-soluble, they can simply slip right through the watery environment of the synaptic cleft. They diffuse backward, and they travel in reverse to bind to specific receptors located on the presynaptic terminal of neuron A.
These are the famous CB1 receptors.
The very same.
And when an endamide, or 2-AG, binds to that CD1 receptor on the shouting neuron A, it acts exactly like a biological circuit breaker. It cuts the power.
It is a remarkably elegant and highly efficient circuit breaker. To look closely at the molecular mechanics for a moment, CB1 receptors belong to a family called G-protein coupled receptors. Specifically, they are coupled to what we call the GI/O subclass.
Okay, let's translate that for a second so we don't get lost in the alphabet soup. When you say it's a G-protein coupled receptor of the GIO subclass, what does that actually mean for the neuron?
The I stands for inhibitory. When the endocannabinoid binds to the CB1 receptor on the outside of the cell, it changes the shape of a protein on the inside of the cell. This triggers a cascade. It inhibits an enzyme called adenyl cyclase, which basically lowers the cell's energy messengers.
It's calming it down.
Exactly. But most importantly, it acts like a bouncer at a club. It blocks the voltage-gated calcium channels from opening, while simultaneously opening the potassium doors to let positive charge out.
And if we remember from a minute ago, calcium is the final trigger that forces the neuron to release its chemical messengers. So no calcium coming in means absolutely no neurotransmitters can be released. Neuron A is effectively silenced. The shouting stops, the circuit breaker flips, and the brain city is protected from overstimulation.
Exactly. And then, almost as quickly as they were created, these natural endocannabinoids are swept away. They are broken down by enzymes like FAAH, fatty acid amide hydrolase. They do their job, they restore the balance, and they vanish within fractions of a second. It is a localized, temporary, and highly precise system of self-regulation.
Which brings us to the core of this entire deep dive, the THC hack. If the brain's natural system is so elegant and localized, how does the primary psychoactive component of a plant fit into this delicate, precisely timed system?
THC is what pharmacologists call a partial agonist at these exact same CB1 receptors. Its molecular structure happens to mimic the brain's natural endocannabinoids just perfectly enough to turn the key in the lock.
But the delivery method is entirely different, right? The brain's natural molecules, 2-AG and anandamide, are released at a single specific synapse for a fraction of a second, exactly when a specific circuit is overheating. But when someone consumes cannabis, THC enters the bloodstream and floods the entire city simultaneously.
That is the critical distinction. It doesn't wait for a neuron to become overexcited. It doesn't target just the synapses that need regulating. It binds indiscriminately to CB1 receptors everywhere in the brain all at once.
And because it's a foreign molecule, it isn't broken down quickly by those cleanup enzymes like FAAH.
No, it isn't. It stays locked into the receptors for hours.
So it's like someone broke into the city's power grid and just started randomly pulling every circuit breaker they could find.
Precisely. By binding everywhere at once, THC causes a widespread, uncoordinated suppression of neurotransmitter release. You see, the brain relies on a very specific signal-to-noise ratio to function. It needs certain circuits to be actively firing and others to be completely quiet so that clear information can be processed. THC effectively flattens this ratio. It muddies the waters.
To really understand what that flattening means for you, the listener, we need to connect this microscopic mechanism to the actual subjective experience.
Yeah.
If we take a tour of the brain on THC, we can see exactly why the high feels the way it does. Instead of just listing off brain regions, let's look at how this widespread hacking alters your reality chronologically.
That's a great approach. Let's start with the cerebral cortex.
Right, the cortex is packed with CB1 receptors. This is the outer layer of the city, responsible for your complex, higher-order thinking and sensory perception. When THC hits here, sensory inputs get scrambled. Colors might seem brighter, music might sound more profound, but your executive function takes a massive hit.
Yes, the cortex is where you process the immediate world. But the scrambling of the cortex alone doesn't explain one of the most famous effects of cannabis: the sudden loss of train of thought. Have you ever been talking to someone who has consumed cannabis and right in the middle of a sentence they just stop and say, wait, what was I just talking about?
All the time. It's like the previous 10 seconds were just wiped from a hard drive.
That phenomenon takes us deeper into the brain, to the hippocampus, which is critical for memory formation. The sources detail how the hippocampus relies on highly synchronized electrical patterns called sharp wave ripples and theta oscillations.
Oscillations, like a rhythm.
Think of these as the rhythmic ticking of a clock that allows the brain to encode short-term memory and establish the sequential order of events.
And when THC binds to the CB1 receptors in the hippocampus, it profoundly disrupts these oscillations. It essentially shuts down the recording equipment. So it impairs the encoding of memory so severely that the user loses what are called their temporal anchors, the sequential markers of what just happened. You forget the beginning of the sentence because the hippocampus never successfully hit save on the concept.
Exactly. And as the user is trying to navigate this scrambled memory, they might also notice a change in their physical coordination. This takes us to the cerebellum and the basal ganglia, the brain's motor control and timing centers.
Right, flooding these areas with THC impairs balance and fine motor skills. The sources highlight some absolutely fascinating research on something called classical eye blink conditioning to prove this point.
Yes, let's talk about the eye blink conditioning. This is a brilliant experimental model. Classical eye blink conditioning is a very specific type of learning dependent entirely on the cerebellum. A subject is trained to blink in response to a tone that predicts a puff of air to the eye.
So the tone plays and they learn to blink right before the air hits.
Exactly. It requires precise microscopic timing. Studies show that chronic cannabis users exhibit markedly fewer and much more poorly timed conditioned responses compared to non-users.
They just can't coordinate the timing of the blink to match the tone.
Right. And what's amazing is that this maps perfectly onto findings in mice that have had their CB1 receptors genetically knocked out. It shows a literal impairment in the circuitry that calculates timing and motor response directly linked to the endocannabinoid system.
Okay, so your thoughts are a bit scrambled, your memory is dropping frames, your physical coordination is slightly delayed, and then invariably you find yourself scaring into the pantry. Let's talk about the munchies. Because suddenly a stale bag of tortilla chips seems like a culinary masterpiece. Why does THC do that?
That takes us to the hypothalamus, the brain's energy regulation center. Normally, when you've eaten enough, the hypothalamus releases a peptide called cholecystokinin, or CCK. CCK is the chemical signal for satiety. It tells your brain, we are full, stop eating. THC actually inhibits the release of CCK. It silences the fullness signal while simultaneously stimulating the pro-appetite pathways. You aren't just psychologically craving food, your brain is being chemically instructed that you are starving.
That is wild. The circuit breaker shuts off the I'm full alarm. But there is another, much more serious emotional effect we have to cover. The amygdala, the brain's emotional core.
This is a critical one.
THC's interaction here is incredibly complex. For some, it causes profound relaxation and euphoria. But for others, or particularly at high doses, it can trigger intense anxiety, fear, and paranoia. Why is it so unpredictable?
It comes back to the fact that CB1 receptors are located on both excitatory glutamate neurons and inhibitory GABA neurons. In the amygdala, THC can inhibit the release of GABA, which normally keeps fear responses in check.
So GABA is usually calming the amygdala down.
Exactly. If you turn off the inhibitory brakes in the fear center, you get runaway anxiety and paranoia. It's a complex disruption throwing emotional processing entirely out of balance, and it varies heavily based on the individual's baseline neurochemistry and the dose of THC.
It really is the widespread, indiscriminate nature of this receptor activation that makes the THC experience so multifaceted. However, if we want to understand why cannabis is inherently rewarding, why people seek it out despite the memory impairments and the potential paranoia, we have to look closely at the dopamine connection. Dopamine is the system that assigns value, motivation, and salience to our actions.
It is the currency of reward, and this is vital to understand.
Here's where it gets really interesting, because the way THC triggers dopamine isn't direct. It isn't like other substances that just force dopamine out. It's a game of neurobiological dominoes. Let's look at the mesolimbic reward pathway, which is the brain's primary reward circuit, connecting the ventral tegmental area, or VTA, down in the midbrain, to the nucleus accumbens in the forebrain.
To use your Metropolis analogy, the VTA is the power plant generating the reward signal, and the nucleus accumbens is the downtown center receiving it. In a normal baseline state, the dopamine-producing neurons in the VTA are kept under incredibly tight control.
They aren't just firing wildly.
Not at all. There are local interneurons right next to them that constantly release GABA, the brain's main inhibitory neurotransmitter.
So these GABA neurons are acting like a constant brake pedal on the dopamine neurons. They keep the dopamine power plant idling, firing at a very low, steady rate of about 1 to 5 hertz, just enough to keep you functioning, but not enough to make you feel euphoric.
Precisely. But those GABA interneurons are absolutely covered in CB1 receptors. So when THC floods the VTA, it binds to the CB1 receptors on those GABA neurons and activates the circuit breaker. The release of GABA is immediately stopped.
By inhibiting the inhibitor, THC effectively takes the foot off the brake pedal, or literally cuts the brake lines. And without that GABA brake, the dopamine neurons are completely disinhibited. They start firing rapidly in high-frequency bursts, flooding the nucleus accumbens with massive waves of dopamine.
This surge of dopamine in the nucleus accumbens creates a powerful sense of reward. It signals to the brain that whatever experience is happening right now has extraordinarily high motivational value. This drives the reinforcement loop that can lead to continued habitual use.
But the dopamine story doesn't end there, does it?
No, it doesn't. What is truly groundbreaking in recent research is the realization that the reward story isn't confined entirely to the midbrain. There is another critical mechanism at play involving the cortex and completely different type of brain cell altogether, the astrocyte.
Yes, astrocytes. These are star-shaped glial cells. And for the longest time, scientists used to think they were just the structural support of the brain. The word glia literally comes from the Greek word for glue. We thought they just had the neurons in place.
That was the dogma for decades. We thought neurons did all the thinking and astrocytes just swept the floors. But recent findings have completely blown that open. Astrocytes are incredibly active participants in neural signaling. And in the cortex, THC activates CB1 receptors on these astrocytes.
But wait, this is where it flips, right?
Yes. Here's the massive paradigm shift. Unlike the CB1 receptors on neurons, which usually couple to those GIO proteins to act as a circuit breaker and inhibit activity, the CB1 receptors on astrocytes uniquely couple to a different protein, GQ proteins.
Okay, let's translate that again. While the neuron's receptors are acting like a brake pedal via GIO, what is this GQ coupling doing on the astrocyte? Is it acting like a gas pedal?
That is exactly right. When THC binds to these astrocytic receptors, the GQ coupling triggers intracellular calcium transients, massive sweeping waves of calcium inside the astrocyte itself, and this calcium surge prompts the astrocyte to actively release glutamate, the brain's primary excitatory neurotransmitter, right into the surrounding synapses.
So instead of shutting things down, the astrocyte is throwing fuel on the fire.
Literally. This burst of glutamate from the astrocytes further activates the frontal corticostriatal pathways. It adds this intense, stimulating layer to the rewarding effects of the drug. The city isn't just turning off traffic lights, it's rerouting massive amounts of power to specific, highly excitable districts.
And this overwhelming flood of dopamine, combined with this highly abnormal release of astrocytic glutamate, leads to one of the most bizarre and universally reported side effects of cannabis consumption.
The time distortion.
The illusion of time, exactly. I'm sure you, listening to this, have either noticed this yourself or heard a friend talk about it. Under the influence of cannabis, time seems to stretch out endlessly. A minute sitting on the couch can feel like an hour. The song you were listening to feels like it has been playing for 20 minutes. Have you ever wondered why that is? The slowing of time isn't just a subjective philosophical illusion, it is a literal mechanical disruption of the brain's timekeeping machinery.
Time distortion under THC is driven by a triad of neurobiological failures. 3 distinct systems breaking down simultaneously. First, as we discussed earlier, the basal ganglia loses its internal rhythm because its circuitry is heavily suppressed by THC. The brain's internal metronome is literally ticking slower.
Okay, so the clock is physically broken. What's the second failure?
The massive increase in dopamine. Dopamine governs salience, how important something feels. When dopamine floods the system, it makes each individual moment feel hypersalient and intense. It expands the subjective weight of the present moment. You are feeling so much in a single second that your brain assumes more time must have passed to contain all those feelings.
And the third failure brings us back to the hippocampus, which is failing to encode short-term memory.
Correct. Because your brain isn't recording the sequence of events properly, you lose those temporal anchors. You can't remember the beginning of your own thought, let alone the context the last 5 minutes.
So when the top-down part of your brain tries to calculate how much time has passed, it looks for memory anchors, finds none, feels the massive emotional weight at the present moment via dopamine, and relies on a broken basal ganglia metronome. The result? It overestimates time wildly. The internal clock is completely out of sync with reality, and that is just staggering to me. It really shows how fragile our perception of reality truly is.
It does. And the profound nature of these acute effects naturally raises a critical question for researchers and users alike. What happens when the brain is exposed to this hack chronically? The brain is a highly adaptive system. It will not simply tolerate a constant state of massive disinhibition, receptor flooding, and temporal distortion. It attempts to defend its homeostasis at all costs, leading to significant neuroadaptations.
Right. The city fights back against the hack. The most immediate defense mechanism the brain employs is called CB1 downregulation. To protect itself from this constant, overwhelming overstimulation from THC, the brain literally removes the CD1 receptors from the surface of its neurons.
It hides them.
Exactly. It puts the traffic lights inside the buildings where the THC can't reach them. The sources you provided highlight some incredible imaging studies showing exactly this.
Yes, these are high-resolution PET scan studies. PE stands for positron emission tomography. In these studies, researchers use a very specific radioactive tracer, or radioligand, called 11C-OMR. This tracer binds specifically to CB1 receptors, allowing us to physically see receptor density in a living human brain.
And what did these scans reveal about chronic daily cannabis users?
They show that chronic daily users have a 15% lower CB1 receptor availability compared to healthy non-users, particularly across the cortical regions. The receptors simply aren't there on the surface anymore.
If we connect this to the bigger picture, a 15% reduction across the cortex is a massive structural shift. And the adaptations don't stop with the endocannabinoid system, do they? The brain also takes aim at the reward system. Because the mesolimbic pathway is being artificially surged by THC, the brain reduces its baseline dopamine production and downgrades its dopamine receptor sensitivity. Over time, those massive dopamine surges start to blunt.
The system builds a tolerance. This creates what we call a hypodopaminergic state, a state of abnormally low dopamine function. And this biological reality maps perfectly onto the psychological symptoms we see in cannabis use disorder.
When the dopamine system is turned down, the user experiences anhedonia. It's often described as an amotivational state. Everyday natural rewards that used to bring joy, like eating a good meal, getting exercise, achieving a goal at work, or just hanging out and laughing with friends, suddenly feel completely flat and uninteresting.
The brain has adjusted its baseline so drastically that only the drug can provide a sense of normal motivation or pleasure.
It is a chemical numbing of the human experience. And beyond just receptor changes, there's evidence of physical, structural damage with chronic heavy use. The animal models detailed in our sources demonstrate actual physical shrinkage of the dopamine-producing neurons down in the VTA.
Yes. Furthermore, there is a documented loss of dendritic spines on the medium spiny neurons within the nucleus accumbens.
Dendritic spines, those are the tiny branch-like protrusions where neurons actually receive incoming signals from other cells, right?
Exactly. They are the physical connection points. Losing them means the network is literally losing its connectivity. The wires are fraying.
And this loss of connectivity creates a massive cognitive conflict within the individual. Your brain normally operates in a careful balance between top-down executive control and bottom-up impulses. The prefrontal cortex, or PFC, is the top-down manager, the mayor of the city. It handles reflection, impulse control, emotional regulation, and long-term planning.
And the basal ganglia and reward centers are the bottom-up drivers of immediate desire and habit. Chronic THC use visibly reduces the volume of the orbitofrontal cortex, a key part of the PFC, and creates what is termed a hypoglutamatergic state.
Glutamate, as we discussed, is the primary excitatory neurotransmitter. It is essential for the rapid, highly complex firing required for top-down executive control. When the PFC is deprived of glutamate signaling, the top-down manager, the mayor, is effectively asleep at the wheel. The individual loses the neurological ability to step back and reflect on their actions.
This drives highly impulsive behavior. It creates a powerful bottom-up urgency to use the drug to escape any negative emotions. Why? Because the very reflective circuitry required to regulate those emotions without the drug has been structurally weakened. It becomes this vicious cycle where the very tool the brain uses to say no is the tool being dismantled by the drug itself.
It is the defining trap of addiction. And this leads us to the critical threshold. What actually happens within the brain when the drug is finally taken away?
It's a profound transition.
There's a moment many people describe when they stop using cannabis. It's not dramatic, it's not loud, it's actually very quiet.
Yeah. And that quiet can feel strange, because for a long time the brain has been receiving the signal from THC.
When someone stops, the brain suddenly has to operate without that signal. The dopamine spikes are smaller, motivation can feel lower, even simple things may feel flat.
And that moment is where many people think something is wrong with them.
But often it's simply the brain recalibrating.
The brain is incredibly adaptive. When THC repeatedly activates the endocannabinoid system, the brain adjusts.
And when the signal disappears, the system slowly learns to balance again.
It doesn't happen instantly, but the brain begins rebuilding its natural dopamine rhythm.
And that's something fascinating about the brain. It can adapt to substances, but it can also adapt to freedom from them.
Which means the story doesn't end when someone quits cannabis.
In many ways, that's where the brain's real recovery story begins.
The biological reality behind that quiet we just described is intensely complex, and it isn't always peaceful. What is happening during this initial phase of abstinence is best explained by the opponent process theory of addiction.
Right. When the brain is chronically exposed to a drug, it pushes back. It mounts biological countermeasures to oppose the drug's effects and try to maintain its homeostasis. When the individual suddenly stops using, the drug is removed. But the brain's massive pushback is still happening. Only now it is completely unopposed by the drug.
This unopposed pushback is what triggers cannabis withdrawal syndrome, or CWS. For decades, there was a myth that cannabis didn't cause physical withdrawal. We now know that is categorically false. It isn't just a psychological desire for the drug. It is a profound physiological crisis of adaptation. The brain's central stress response system, known as the HPA axis, becomes highly dysfunctional.
Wait, before we talk about the panic of withdrawal, let's define the HPA axis for a second so everyone is on the same page. That stands for the hypothalamic pituitary adrenal axis, right? Basically, it's the body's central alarm system that controls how we respond to stress.
Yes, it is the feedback loop that dictates your stress levels. And without the suppressive, dampening effects of THC that the brain had grown accustomed to, this HPA axis goes into overdrive. The brain releases a massive surge of corticotropin-releasing factor, or CRF.
And CRF is the brain's primary stress chemical. This massive surge is responsible for the intense anxiety, the profound irritability, the vivid nightmares, and the severe sleep disturbances that define the acute withdrawal phase.
The brain is essentially in a state of chemical panic. It is trying to operate a system that has had its primary inhibitory receptors, the CB1 receptors, downregulated and hidden away, while simultaneously dealing with blunted dopamine levels and a flood of stress hormones. It feels awful because biologically the system is temporarily broken.
But the incredible news, and the real hope within the research you gathered, is the timeline of cognitive restoration. The brain possesses a staggering capacity for neuroplasticity. The recalibration process is arduous, certainly, but it is highly effective. Let's look at the early phase of recovery, spanning days 1 through 28. The speed at which the brain begins to heal its endocannabinoid system is nothing short of remarkable.
It really is. Remember those PT scans using the 11C OMAR tracer that showed the 15% downregulation in chronic users? Researchers used those same scans to track recovery. Within just 48 hours of monitored abstinence, the scans show a significant, visually measurable upregulation of CB1 receptors.
2 days. That's all it takes for the brain to realize the flood is over. The brain immediately senses the absence of the exogenous chemical and begins pushing those receptors back up to the surface of the neurons to capture whatever natural endocannabinoids it can produce. It's putting the traffic lights back on the streets.
Exactly. And within the first week, many individuals report that the mental fog begins to lift. Clinical research shows that verbal learning and memory capacity demonstrate dramatic improvement during these first 7 days.
And this correlates perfectly with the residual THC finally washing out of the fat-rich tissues of the hippocampus, right? Once the THC is gone, those sharp wave ripples and theta oscillations, the brain's internal recorders, can begin syncing up again. The temporal anchors return.
By day 28, the receptor density in the vast majority of cortical regions has returned to levels that are statistically comparable to healthy, non-using individuals. At this 4-week mark, the acute physical withdrawal symptoms driven by that terrible CRF stress surge have largely dissipated. The alarm system has turned off. However, it is vital to note that the hippocampus may take a bit longer to fully upregulate its receptors, which explains why some subtle memory issues can linger past the first month.
So what does this all mean for the person going through it? It means that the physical hardware, the CB1 receptors themselves, heal incredibly fast. But the software, the delicate rewiring of the deeper emotional and decision-making circuits, is a much longer journey. This brings us to the intermediate and long-term recovery phase, from month 2 up to a year.
While the receptors return to the surface quickly, the underlying synaptic plasticity, the actual strength and health of the connections between neurons, takes significantly longer to repair. The deficits in complex executive functions, sustained attention, and inhibitory control, those top-down prefrontal cortex functions we discussed, can endure for 3 to 4 weeks and often take up to 2 or 3 months to fully normalize.
The mayor needs time to get back into the office and start managing the city again. The brain literally has to rebuild the glutamatergic pathways that allow for reflective thought and impulse control.
It is a gradual rebuilding process, and then there is the dopamine system. The recovery of the dopamine baseline is what finally cures that heavy, quiet flatness we talked about in the opponent process phase. By month 2 or 3, the dopamine system begins to genuinely normalize. The nucleus accumbens slowly regains its sensitivity.
It begins responding effectively to natural rewards again. The joy of going for a run, the pleasure of a deep conversation, the emotional engagement with a piece of music. These natural reinforcers start to feel genuinely good again because the dopamine circuitry has finally shed the blunting effects of chronic use. The brain doesn't need the massive artificial flood to feel joy anymore. It can appreciate the gentle natural tides again.
It is a profound testament to the resilience of the human central nervous system. The brain can completely re-architect its reward and signaling pathways to accommodate a massive chemical intrusion, and then, given enough time and abstinence, it can systematically dismantle those exact same adaptations to restore its original healthy homeostatic balance.
Let's bring this all together. We have journeyed from the tiny electrical spark of a single action potential, relying on the tireless, energy-hungry work of the sodium-potassium pump, to the elegant circuit breaker of retrograde signaling via the endocannabinoid system. We've seen how a single molecule, a molecular master key called THC, can hijack this system. We watched it flood the locks, inhibit the GABA brakes, and surge dopamine, all while simultaneously distorting our very perception of time by disrupting the basal ganglia and the hippocampus.
We tracked the heavy, sometimes hidden toll of chronic exposure, the downregulation of receptors, the blunting of the reward system, and the physical pruning of dendritic spines that leads to intense cognitive conflict and impulsivity. But we also mapped the beautiful, complex machinery of recovery, watching receptors reemerge in a mere 48 hours and dopamine systems reawaken over a span of months.
It is a profound story of chemical manipulation and deep biological resilience. And it leaves us with something quite incredible to think about regarding how our minds actually construct reality.
Indeed. Our brains possess this ancient, highly intricate endocannabinoid system entirely dedicated to keeping our physical and emotional reality in a state of perfect balance. If a single plant molecule can so profoundly hijack this system to completely alter our perception of time, our encoding of memory, and our capacity for joy, what other hidden master regulators exist within us, silently constructing the reality you are experiencing right now, just waiting to be understood.
Keep questioning the world inside your own head. The architecture of your reality is more complex, more adaptable, and more fragile than you could ever imagine. Thank you for joining us on this deep dive. Imagine a system hidden inside your brain, a system designed to control mood, motivation, memory, even pain. Now imagine a plant chemical that can plug directly into that system.
And when it does, the brain begins to change how it creates pleasure. This chemical is THC, and the system it hijacks already exists inside you.
Welcome to this deep dive. I'm looking at this massive stack of neurobiological research papers right now, medical journals, clinical studies we've gathered for you today. And, well, the sheer volume of data is just staggering.
It really is. There's so much to get through.
Yeah. Our topic today is how cannabis, specifically THC, hacks the brain. But to be clear right off the bat, we aren't just looking at the surface level effects here.
No, not at all.
We are bypassing the standard conversations entirely. Our mission today is to extract the most fascinating, mind-bending insights from these scientific papers and translate them into a clear map of the molecular mechanics happening right inside your head.
Right.
We're talking about the architecture of reward, the physical distortion of your perception of time, and the actual neuroadaptation of your cells.
Which is really the only way to look at it if you want to understand what's actually going on. Because the deeper you look at the pharmacology here, the more you realize that the story of cannabis isn't really a story about a plant. It's a mirror reflecting human neurobiology. To understand the external chemical, you really have to deeply understand the pre-existing internal architecture.
Let's start right there, because I think there's this common misconception that cannabis simply acts on the brain to make you relaxed or hungry, almost as if the plant itself is generating those feelings out of thin air.
Yeah, like it's some kind of magic.
Exactly. But the reality is that the story begins inside a biological network that evolution installed inside of you long before humans ever even discovered the cannabis sativa plant.
Long before.
We're talking about the endocannabinoid system. Okay,
Yeah.
let's unpack this, because before we get to the hack, we need to understand the system that's being hacked.
Right, and the evolutionary timeline of this system is just incredible. The endocannabinoid system, or ECS, it actually emerged before the evolution of vertebrates. Yeah. It is remarkably well preserved across totally diverse species, from like sea squirts all the way up to mammals. Your brain, right now, as you are listening to this, is actively producing cannabinoid-like molecules.
I was actually looking into the methodology of how we even figured this out, because the timeline is, well, it's a bit backward.
It is backward.
The main psychoactive component of marijuana, delta-9 tetrahydrocannabinol, also THC,
Yeah.
was isolated back in 1964 by researchers Raphael Mechoulam and Yekil Gaoni. But reading through the historical context, it seems like for decades after isolating THC, scientists had absolutely no idea how it actually functioned in the brain.
They really didn't. I mean, for a long time, the prevailing theory was that THC just had a non-specific effect on neuronal cell membranes.
Just kind of washing over the brain.
Yeah, exactly. Because it's highly lipophilic, meaning it loves fat, they assumed it just sort of dissolved into the fatty membranes of neurons and altered their function in a very generalized, clumsy way. Similar to how we used to think general anesthetics worked. It wasn't until 1988 that researchers using radio-labeled synthetic cannabinoids finally discovered a highly specific receptor in the brain that THC binds to.
And that discovery must have just set off alarm bells in the neurobiology community, because nature doesn't evolve a highly specific, complex receptor in the mammalian brain just waiting millions of years for a specific plant to come along.
No, it definitely doesn't.
If we have a receptor for it, we must make our own version of it.
That was exactly the hypothesis. It launched this massive scientific hunt for the endogenous ligand, which is just the internal molecule that naturally fits that receptor. And that hunt finally culminated in 1992 when a researcher named William Devane, working in Mechoulam's lab, isolated the first endocannabinoid.
And the methodology for that was intense, wasn't it?
It was grueling. They were processing massive amounts of porcine brains, so pig brains, using mass spectrometry and nuclear magnetic resonance spectroscopy just to isolate this tiny, elusive lipid molecule.
And when they finally isolated it, the name they chose is just fantastic.
I love the name.
They called it anandamide, coming from the ancient Sanskrit word ananda, which translates to internal bliss or extreme happiness.
It's very poetic for a biochemical compound, isn't it?
It really is.
And shortly after that, scientists identified a second major endocannabinoid called 2-arachidinyl glycerol, or 2-AG. Now, it's critical to understand what these molecules actually are.
Right.
Because unlike classic neurotransmitters like serotonin or dopamine, which are synthesized ahead of time and stored in little cellular vesicles waiting to be released.
Like little storage units.
Exactly. Anandamide and 2-AG are different. They are bioactive lipids. They are fat-based molecules derived from arachidonic acid in the cell membrane itself, and their function is vastly more complex than just generating bliss.
They do a lot more than that.
They are the ultimate biological regulators. They govern stress recovery, mood regulation, memory consolidation, appetite, immune response, and even pain sensation.
But the way they regulate is what really caught my attention in these papers, because it completely breaks the standard model of neuroscience.
It really throws a wrench in the old textbooks.
Right, because for anyone who follows neurobiology, we are used to a very specific direction of traffic. A signal travels from a presynaptic neuron, the sender, across the synapse, to the postsynaptic neuron, the receiver. But the endocannabinoid system operates via retrograde signaling.
Yes.
They go backward.
They do, and it was a massive paradigm shift when researchers realized this. For a century, we thought the brain was strictly a one-way street, but the endocannabinoid system proved the brain can talk backward.
So how does that work in practice?
Well, to visualize this, let's look at how a neuron handles being overstimulated. Let's say a postsynaptic neuron, the receiver, is getting an overwhelming barrage of excitatory signals. It's getting too noisy.
Okay, too much input.
Right. That excessive calcium influx inside the receiving neuron triggers specific enzymes, like DAG lipase, to rapidly synthesize endocannabinoids on demand directly from the lipid membrane.
So they aren't stored up at all. They are manufactured right in the heat of the moment.
Precisely. The moment they're synthesized, they slip out of the postsynaptic neuron, travel backward across the synaptic cleft, and bind to receptors on the presynaptic neuron.
The sender that was making all the noise in the first place.
Exactly.
I'm trying to think of a good way to conceptualize this for you listening. It's less like a standard lock and key and more like an active noise-canceling algorithm in a pair of high-end headphones.
Oh, that's a great way to put it.
The headphones listen to the incoming noise from the environment, and they instantly generate a counter signal to suppress that specific frequency, creating quiet. The endocannabinoids are the brain's biological noise-canceling system. They travel backward, hit the presynaptic neuron, and basically say, hey, we've got the message, stop releasing neurotransmitters.
That is a highly accurate analogy. In the literature, you'll see this referred to with terms like depolarization-induced suppression of excitation, or DSE, or if they're suppressing inhibitory signals, DSI.
We don't need to get bogged down in the acronyms, though.
No, you don't. The core insight here is just that this retrograde signaling allows the brain to finely tune the balance of excitation and inhibition across these massively complex neural circuits. It is the master volume knob for homeostasis.
And to turn that volume knob, these endocannabinoids bind to specific receptors. The sources highlight two main types, CB1 and CB2. Let's focus heavily on CB1 because the data on its distribution is just wild.
It really is. CB1 receptors are G-protein-coupled receptors. Now, without getting too lost in the weeds, a G-protein-coupled receptor doesn't just open a simple channel to let ions flow in.
It's more complicated than that.
Much more. When a molecule binds to it, it triggers a complex cascade of intracellular events, almost like a molecular Rube Goldberg machine, that ultimately alters the cell's behavior. What's staggering is that CB1 receptors are among the most incredibly abundant G-protein-coupled receptors in the entire vertebrate brain.
They are everywhere.
They are packed densely into the cerebral cortex, the hippocampus, the basal ganglia, and the cerebellum.
While the CB2 receptors are mostly localized in the peripheral immune system, though some recent studies in our stack found them in brain regions, like the ventral tegmental area, modulating neuroinflammation.
Right, but the CB1 receptor is the main player for the psychoactive effects.
So, bring us back to you, the listener. Imagine the architecture inside your head. Spread across billions of neurons are these specialized CB1 receptors, highly concentrated in areas controlling memory, movement, and executive function.
Just waiting.
They're waiting for your natural anandamide to slide in, run the noise-canceling algorithm, and keep your cognitive environment perfectly balanced.
The brain already has the receptor network fully integrated and operational.
And THC is a master key that fits those exact receptors.
Exactly. When we talk about THC hacking the brain, we are looking at the consequences of an external molecule forcefully inserting itself into this delicate retrograde signaling system.
So let's track the invasion. Let's visualize the exact physiological moment of exposure.
Okay, let's do it.
Someone consumes cannabis, and the THC enters the bloodstream. It races up toward the brain and hits the blood-brain barrier. Now, the blood-brain barrier is a highly selective fortress. It normally keeps most toxins and external chemicals out.
It's very good at its job.
But because THC is incredibly lipophilic and the barrier is largely lipid-based, THC slides right through the gates, completely undetected.
It is a stealth molecule in that regard. Once inside the central nervous system, the THC begins binding to those widespread CB1 receptors. But here's where the concept of the hack really crystallizes, because THC's interaction with the receptor network is actually quite clumsy compared to the brain's natural endocannabinoids.
I read that and had pause for a second, because clumsy isn't the word most people would associate with a chemical that produces such profound subjective effects. If anything, the effects feel incredibly potent to the user.
They do, but the profound nature of the effect is directly caused by that clumsiness.
How so?
We have to look at spatial and temporal specificity. Natural anandamide is incredibly precise. It is synthesized on demand at a specific synapse, travels backwards, suppresses the noise, and then it is immediately destroyed.
Right away.
Yes. The brain deploys an enzyme called FAH, fatty acid amide hydrolase, which acts as a biological cleanup crew. Within minutes, often even seconds, of anandamide doing its job, FAAH breaks it down. It's a highly targeted, localized signal that vanishes as quickly as it appears.
It's like a targeted text message that self-destructs the moment it's read.
Exactly. THC, however, operates completely differently. First, it is highly resistant to that enzymatic breakdown. FAAH cannot effectively dismantle the THC molecule, so it lingers in the system for a vastly longer period.
And the second difference.
Second, because you are introducing it externally via the bloodstream, it doesn't act locally at a single noisy synapse. It floods the entire brain simultaneously, binding indiscriminately to nearly all available CB1 receptors at once. It has zero spatial or temporal specificity.
So it's not a targeted text message at all. It's a massive radio broadcast, overriding every single communication channel in the brain simultaneously.
That is the perfect way to look at it.
And because it's overriding these receptors everywhere all at once, the brain's communication system becomes physically distorted. And the fascinating part is that we can actually map the subjective experiences of being high directly to the specific brain regions being flooded.
We can trace it with remarkable accuracy now? Take memory, for instance.
Let's look at memory.
We mentioned earlier that the hippocampus is densely packed with CB1 receptors. The hippocampus is your brain's memory sorting center. It is responsible for encoding short-term memories and spatial navigation.
Okay.
When THC floods the hippocampus, it suppresses the normal firing patterns of the neurons there. They can't synchronize their oscillations. The result is that short-term memory encoding is profoundly disrupted. This is why a user might lose their train of thought mid-sentence. The hippocampus literally failed to lay down the memory of how the sentence even began.
And the physical relaxation, or the impairment of fine motor control, that maps directly to the basal ganglia and the cerebellum.
Exactly.
The basal ganglia are heavily involved in initiating movements, while the cerebellum handles the fine-tuning of balance and coordination. Flooding these areas alters the firing of the neurons, managing these processes, leading to that characteristic heavy-limbed feeling.
But one of the most fascinating distortions, I think, occurs with time perception.
Yes, the time dilation effect. Why does it feel like a single minute is dragging on for an hour? The clinical studies provided in our sources dive incredibly deep into this, specifically looking at the science of interval timing.
Interval timing, right?
That's how we perceive time in the seconds to minutes range. To the brain, time perception isn't controlled by a single, centralized ticking clock. It's a distributed network involving memory, attention, and internal rhythmic pacemakers.
And the methodology of these interval timing studies is quite rigorous. Researchers administered intravenous THC to volunteers in double-blind, placebo-controlled settings. They used highly specific doses, ranging from 0.015 to 0.05 milligrams per kilogram, just to ensure totally accurate blood plasma levels.
And then they tested them.
Then they had the subjects perform time estimation and time production tasks.
Just to clarify the difference there, because I found the study design really clever. Time estimation is when the researcher plays two beeps and asks, how much time just passed between those sounds?
Great.
Time production is when they hand the subject a stopwatch and say, stop this when you think exactly 60 seconds have gone by.
Right. And the data from these IV THC studies is incredibly consistent. THC reliably causes profound time overestimation and time underproduction.
Let's break those down.
Time overestimation means the subject thinks vastly more time has passed than actually has in reality. So if 30 seconds pass, they might estimate it was 90 seconds. And time underproduction means their internal clock is running so fast that if you ask them to wait a full minute, they'll hit the stopwatch at 35 or 40 seconds, genuinely believing a full 60 seconds has elapsed.
So their internal subjective time is accelerating, which makes external reality feel like it is crawling in slow motion. But what is the THC actually doing to the physical machinery of the brain to cause that dilation?
In the neurobiological literature, temporal processing is divided into a clock stage, a memory stage, and a decision stage. THC directly impacts the basal ganglia, which houses a massive population of medium spiny neurons.
Medium spiny neurons.
Yes. These neurons act as the brain's internal pacemaker. They integrate motor signals and temporal information. When THC binds to the CB1 receptors in the basal ganglia, it alters the dopamine and glutamate transmission there, essentially accelerating the rhythm of these medium spiny neurons. Your internal metronome starts ticking at double or triple speed.
But I read that the memory disruption in the hippocampus also plays a critical role in this illusion.
It's the combination of the two that makes the effect so profound. Normally, you judge the passage of time by the sequence of events, thoughts, and sensory inputs you've recorded in your short-term memory over the last few minutes. They act as temporal anchors.
Makes sense.
But because THC is simultaneously disrupting the hippocampus, those recordings are fragmented. You are generating more mental frames per second because your basal ganglia metronome is ticking faster, but your hippocampus isn't anchoring those frames to a continuous context.
Let me use an analogy here that I think everyone listening can relate to. Have you ever stood in front of a microwave waiting for something to heat up for 60 seconds?
Oh, absolutely. It takes forever.
You just stare at the timer. Because your attention is hyper-focused and you aren't laying down any other contextual memories, you're just standing there. Those 60 seconds feel like an eternity. With THC, the brain is chemically forced into a similar state. The metronome is racing, generating tons of mental frames, but memory isn't anchoring them. To you, a single minute feels strangely immersive, stretched out, and completely disconnected from the minute that preceded it.
It is a profound sensory illusion, but experiencing dilated time and a lack of short-term memory doesn't inherently explain why people actually seek out the experience.
That's true.
To understand why this state is experienced as rewarding, we have to look at the core of the brain's motivation system. We have to talk about dopamine.
Which brings us to the centerpiece of this deep dive. Dopamine is the primary currency of the brain's reward architecture. And to understand how THC hacks this, we need to look at the mesolimbic dopamine pathway.
The main reward circuit.
This is a vital neural circuit that originates deep in the midbrain in an area called the ventral tegmental area, or VTA, and it projects its nerve fibers up to a region in the forebrain called the nucleus accumbens.
This specific pathway is, essentially, the biological engine of human survival and motivation. Normally, dopamine levels rise in the nucleus accumbens when you accomplish something crucial for your survival or the propagation of your genes.
Give me an example of a natural trigger.
If you solve a complex problem, if you secure a major social victory, if you find calorie-dense food when you are starving, your VTA fires off a burst of dopamine. It's the brain's way of tagging that specific behavior as highly salient. It is a chemical signal that says that was incredibly beneficial. Remember exactly the sequence of actions you took to achieve that, and do it again.
It's the ultimate evolutionary reinforcement mechanism.
It is.
So naturally, drugs of abuse tap into this system. But as I was reviewing the papers on dopamine synthesis for this deep dive, I hit a massive paradox regarding how THC actually interacts with this system.
The paradox is one of the most elegant discoveries in modern neurobiology, honestly. If you look at other substances, cocaine, for instance, it physically blocks the dopamine transporter, allowing dopamine to build up in the synapse. Amphetamines actually enter the neuron and force dopamine out of the vesicles. They interact directly with the dopamine machinery.
They go right to the source.
But the actual dopaminergic neurons in the VTA, the very factories that produce and release the dopamine, generally lack CB1 receptors altogether.
Wait, if the THC master key can't actually fit into the dopamine factories, how does it trigger the massive dopamine surge that users experience? How does it hijack the reward system if it can't even touch the reward neurons?
Because it executes a brilliant indirect double hack utilizing the complex local circuitry surrounding those dopamine neurons.
Okay, play this out for us.
The first mechanism is a process called GABAergic disinhibition. To understand this, we have to picture the VTA dopamine neurons not as running freely, but as being constantly held under strict lock and key. They're surrounded by inhibitory GABA interneurons.
GABA being the main inhibitory neurotransmitter.
Right. GABA is the primary inhibitory neurotransmitter in the brain. These GABA interneurons are constantly releasing GABA onto the dopamine neurons, essentially keeping a biological brake pedal pressed firmly to the floor so that dopamine is only released when truly earned.
Okay, so the brake pedal is pushed all the way down.
Now, while the dopamine neurons lack CB1 receptors, those surrounding GABA interneurons are absolutely covered in them. When THC floods the midbrain, it binds to the CB1 receptors on the GABA interneurons.
And remember, endocannabinoid signaling is a retrograde off switch. It's a noise-canceling algorithm.
Exactly. THC triggers that off switch, forcing the GABA neurons to stop releasing GABA.
It cuts the brake lines.
It cuts the brake lines entirely. By cutting the brake lines, the dopamine neurons are suddenly disinhibited. Without that constant heavy suppression from the GABA interneurons, the dopamine neurons begin to fire spontaneously and rapidly. Their burst activity increases significantly, and they send a massive flood of dopamine rushing down the projections into the nucleus accumbens.
That is just incredible biological engineering. It doesn't push the dopamine neuron. It just chemically removes the guard that was holding it back.
It's a backdoor bypass.
But I found another paper in the stack that suggests that's only the first half of the hack. There's a second mechanism happening simultaneously involving a completely different part of the brain.
Right. Because while THC is cutting the brakes down in the midbrain, it is simultaneously hitting the accelerator from the cortex.
Hitting the accelerator.
This is a much more recent discovery involving astrocytic CB1 activation. Astrocytes are a type of star-shaped glial cell in the brain. For decades, neuroscientists thought astrocytes were just structural support cells, the glue holding neurons together.
But they do more than that.
We now know they are deeply involved in active neurotransmission.
And THC binds to them as well.
It does. THC binds to CB1 receptors located on astrocytes in the prefrontal cortex. Now, unlike neuronal CB1 receptors, which typically inhibit signal release, activating these astrocytic CB1 receptors triggers an intracellular calcium cascade that causes the astrocyte to release glutamate. And glutamate is the brain's primary excitatory neurotransmitter.
The ultimate green light.
Yes. This localized release of glutamate from the astrocytes excites pyramidal neurons in the cortex. Pyramidal neurons are these large, long-range projection neurons. When they get excited by the glutamate, they send powerful excitatory signals all the way back down into the reward center, the striatum, and the nucleus accumbens.
So it's coming from both directions.
So look at the entire picture. You have massive excitatory signals raining down from the cortex into the reward center right at the exact same time that the midbrain dopamine neurons have had their GABA brake lines cut. The combined result of this dual mechanism is a profound artificial surge in dopamine.
Let's put this into a real-world context for you listening. Normally, to get a dopamine surge of that magnitude naturally, you would have to do something extraordinary.
Truly
You
extraordinary.
have to climb a mountain, fall deeply in love, or win a championship. You would have to go through a rigorous natural achievement process. THC completely shortcuts that entire process. It provides an unearned, massive, artificial reward signal.
Unearned is the critical word from a neurobiological perspective. The brain's architecture is registering a massive survival-level success, but the physical body might just be sitting on the couch.
And because dopamine doesn't just make you feel good, but it also dictates salience, meaning it tells your brain what is important in your environment, this artificial surge makes utterly mundane stimuli feel profoundly significant.
The illusion of profundity.
Yes, this explains the illusion of profundity. It's why, when under the influence, staring at the visual patterns of a rug, or listening to a song you've heard 1,000 times, or eating a handful of potato chips, suddenly feels like a deeply rewarding, almost religious experience.
Yeah.
The dopamine is screaming at your brain, pay attention to this is the most important thing happening right now.
The illusion is entirely generated by cutting the brake lines and hitting the accelerator of your mesolimbic pathway simultaneously. The external stimulus isn't actually more profound. Your brain is just being chemically instructed to tag it as profound.
Which is just mind-tending to conceptualize. But this leads us to a highly critical pivot in the research. Because the brain isn't just a passive receiver of chemicals.
No, it's not.
It doesn't just sit there and let its reward system be hacked day after day without responding. The brain is an incredibly dynamic, adaptive system. So we have to ask the inevitable question. What happens when this system is hacked repeatedly? When a person moves from acute occasional use to chronic daily exposure?
This is where we enter the territory of neuroadaptation, and it is crucial to understand the mechanics here. The brain's overarching goal at all times is homeostasis. It aggressively wants to maintain balance.
It wants to keep things normal.
Exactly. It expects a certain baseline level of receptor signaling. THC acts as a partial agonist at the CB1 receptor. A partial agonist is a molecule that binds to a receptor, but only produces a partial response compared to the body's full natural agonist.
Okay.
However, because THC floods the system in such massive concentrations and resists enzymatic breakdown, the net effect is a staggering overstimulation of the entire endocannabinoid network. The volume is turned up way too loud for way too long.
So if the volume is too loud, the brain tries to turn it down itself.
Exactly. To defend itself against this constant chemical bombardment, the brain initiates a cellular process called downregulation. It literally pulls its CB1 receptors away from the surface of the cell membrane. It internalizes them into the cell where they can no longer be activated.
It hides them.
If THC is the master key, the brain's defense mechanism is to physically remove the keyholes so that THC has fewer and fewer places to bind.
It's a brilliant, highly defensive learning machine. But that cellular defense mechanism translates into real-world behavioral consequences for the user. As we mapped out the clinical studies, there are three main consequences of this receptor downregulation that we need to dive into.
Let's go through them.
The first one is tolerance.
Tolerance is the most immediate and noticeable consequence of downregulation. Because the overall density of available CB1 receptors has dropped significantly, the listener needs more and more of the external substance to achieve the same effect they used to get from a smaller amount.
Makes total sense.
A perfect example of this is the time dilation effect we discussed earlier.
Right, I noticed that in the interval timing data.
Yes, the studies show that while infrequent users experience massive time overestimation at medium doses, chronic daily users showed almost no difference in time estimation compared to a placebo.
Oh, wow.
Their brains had downregulated the CB1 receptors in the basal ganglia so heavily that the THC could no longer accelerate the medium spiny neurons enough to distort their perception of time. The brain successfully blunted that specific effect.
It adapted completely. The second consequence is dopamine baseline shifting. And looking at the data, this seems incredibly serious regarding long-term motivation.
It is arguably the most impactful neurobiological consequence. Remember that the mesolimbic dopamine pathway is specifically designed to reward you for natural survival behaviors. But with chronic THC exposure, the system adapts to expect massive artificial floods of dopamine on a regular basis.
It gets used to the shortcut.
Exactly. Over time, the dopamine response to non-drug rewards, things like receiving money, eating a great meal, or having a positive social interaction, becomes severely blunted.
Because the natural rewards simply can no longer compete with the artificial chemical surge. I was looking at the methodology of how we measure this. The sources detailed neuroimaging studies, specifically positron emission tomography or PET scans.
The PE scan methodology is fascinating. Researchers use specific radiotracers, molecules tagged with a radioactive isotope that bind to dopamine receptors or synthesize into dopamine.
Right.
They inject these into the subject and use the scanner to watch the brain light up in real time. These PE studies have conclusively shown that chronic cannabis users have lower dopamine synthesis capacity and a blunted dopamine release in response to stimulant challenges compared to healthy controls.
Meaning the baseline capacity of the entire reward system gets turned down, which leads directly into the third consequence, reduced natural motivation, which clinically presents as anhedonia.
Right. Because the reward center is physiologically desensitized and the dopamine baseline has shifted downward, the user might find it significantly harder to experience pleasure or motivation from everyday accomplishments.
It's just harder to get going.
Anhedonia is the clinical term for the inability to feel pleasure. If natural everyday rewards don't trigger enough dopamine to surpass the new lowered baseline, there is no neurochemical drive to pursue them. The brain has adapted to rely heavily on the external drug just to reach what used to be its normal, sober baseline of feeling okay or motivated.
And it becomes a self-perpetuating cycle. You lack motivation because your dopamine system is blunted by downregulation, so you seek the external drug to feel rewarded or motivated, which then causes the brain to downregulate the receptors even further to defend against the surge.
It's a tight feedback loop.
That is a sobering reality. But it's important to frame this correctly. It's not a moral failure. It is a highly predictable mechanical biological adaptation. Your brain is just doing exactly what it evolved to do to protect itself from overstimulation.
That's entirely true.
But this raises the ultimate question, one that I'm sure anyone listening is wondering right now. Is this downregulation permanent? What happens if the exposure completely stops? Can the brain rebuild the keyholes?
The science here is actually quite encouraging, and again, we rely heavily on PP imaging data to answer this. Researchers use a specific radiotracer, like F18-FMPE-PD2, which binds selectively to CB1 receptors to map the receptor density in chronic daily cannabis smokers, and then scan them again after periods of monitored abstinence.
And what did they find?
The data shows that CB1 receptor downregulation is largely and surprisingly quickly reversible.
How quickly are we talking? What does the timeline look like for the brain to repair the hack?
After about 4 weeks of sustained abstinence, the receptor density in chronic users normalizes almost completely, returning to levels comparable to healthy non-users.
4 weeks?
Yes. Once the brain recognizes that the massive chemical flood has receded, it begins to reverse the defense mechanism. It pushes the CB1 receptors back up to the surface of the cell membrane to reestablish normal endogenous signaling. Now, it's worth noting there are regional differences in this recovery.
Meaning different parts of the brain recover at different speeds.
Exactly. Subcortical regions, like the basal ganglia, which handles that time dilation and motor control, tend to bounce back and upregulate faster than cortical regions, like the prefrontal cortex, which handles complex decision-making.
That makes sense.
But overall, the adult brain's inherent plasticity allows it to repair the hack and reset the locks within roughly a month.
4 weeks to reset the board. That is an incredible testament to the resilience of the human brain. But there is a crucial caveat here in the literature, isn't there? You specifically said the adult brain.
Yes. This is a vital distinction highlighted extensively across the neurodevelopmental papers. The adult brain is highly resilient and plastic in its recovery, but the adolescent brain is uniquely vulnerable.
Why is that? What is fundamentally different about the teenage brain when it comes to THC exposure?
During adolescence, the brain isn't just growing, it is undergoing a massive, critical period of structural reorganization. It is maturing the prefrontal cortex, the area responsible for high-level executive function, decision-making, and impulse control.
It's still under construction.
Exactly. It is also undergoing a vital process called synaptic pruning. The brain overproduces neural connections in childhood, and during adolescence, it aggressively prunes away the weak connections and coats the important ones in myelin, strengthening them. This increases the efficiency of the brain's white matter tracts.
And what role does the endocannabinoid system play in that pruning process?
It is the biological architect. The natural endocannabinoid system actively guides this structural development. Anandamide and 2-AG are the signaling molecules telling the neurons exactly how to wire themselves for adulthood, determining which synapses to keep and which to prune.
So if you introduce heavy, indiscriminate THC exposure during this specific developmental window, you aren't just temporarily turning down the volume on a finished system. You are actively distorting the architectural blueprints of the brain while the house is being built.
That is the exact concern. Animal models and human imaging studies suggest that heavy, chronic cannabis use in early adolescence can lead to enduring structural changes. We see altered white matter integrity and long-lasting cognitive deficits that do not easily reverse after 4 weeks of abstinence because the foundational wiring of the brain was altered during a critical window.
The biological stakes are vastly different depending on the developmental stage of the user. And again, our goal here is to present this data objectively. This isn't about judgment. It is purely a fascinating look at how a highly complex system reacts to external inputs.
Absolutely.
So what does this all mean for you? It means your brain is a brilliant, highly defensive learning machine. It adapts precisely to the environment you put it in and the chemicals you expose it to.
It isn't just reacting in the moment, it is constantly updating its own structural and chemical source code to prepare for the future based on the inputs of today.
Cannabis itself isn't the whole story.
The deeper story is how the brain learns from every single signal it receives.
So the real question isn't just what THC does to the brain, but what happens when the brain learns to expect it.
And if we step back and look at the endocannabinoid system as a whole, it leaves us with one last, truly provocative concept to mull over.
What's that?
We've spent this entire time talking about what happens when you hyperstimulate the system with a massive external flood of plant chemicals. But what if the system is naturally underperforming?
What do you mean? Like a genetic defect?
Exactly. There is a growing body of rigorous medical research exploring a concept known as clinical endocannabinoid deficiency, or CED. Just as some people are born with genetic factors that affect their serotonin production leading to depression, or dopamine production leading to Parkinson's, what if some people are born with a genetic defect that prevents their bodies from producing enough natural anandamide? Or what if they possess mutated, defective CB1 receptors?
Their internal volume knob is fundamentally broken from birth.
Yeah.
The noise-canceling algorithm doesn't work. The brakes are worn out.
Precisely. If anandamide is the master regulator responsible for controlling pain signals, mood stability, and digestion, a chronic deficiency would mean a baseline state of constant hypersensitivity. The brain would be perpetually too noisy.
That sounds awful.
It is. Research suggests that this natural deficiency might actually be the hidden neurobiological root cause of several notoriously difficult-to-treat chronic conditions, things like severe treatment-resistant migraines, fibromyalgia, and irritable bowel syndrome. Patients with these specific conditions often show significantly lower levels of endocannabinoids in their cerebrospinal fluid, or they exhibit genetic variations in the enzymes that synthesize and degrade these lipids.
That reframes the entire conversation around this system, because it forces us to wonder, could the future of medicine lie not in hacking the brain with external plants that flood the whole system indiscriminately, but in developing highly targeted precision therapies to simply repair or gently boost the brain's own internal bliss machinery? What if we could just fix the volume knob instead of blasting the speakers?
It's a tantalizing prospect for the future of neurology. It moves the scientific conversation entirely away from recreational hacking and toward profound, targeted neurological healing. Understanding how the cannabis plant hijacked the system was merely the gateway to understanding the profound capabilities of our own biology.
And what a gateway it has been. From the grueling mass spectrometry of porcine brains to find the molecule of internal bliss, to backward-traveling noise-canceling lipids, cut GABA brake lines, accelerated basal ganglia metronomes stretching the fabric of time, and a brain that literally rebuilds its own cellular locks to survive the flood. We hope this map of the molecular mechanics of your mind has been as mind-bending for you as it was for us to explore. Stay curious, keep questioning the world inside your head, and keep diving deep. Until next time.